Okay. I guess it's an afternoon, so good afternoon. Thank you all for attending our pre-meeting workshop on what to expect as a participant in a gene therapy clinical trial. Just as a reminder, before we get started, that this is an in-person and also a virtual session, so thank you guys all who are dialing in as well. So as a reminder, a couple housekeeping items. Please silence your phones and mobile devices. Also, please refrain from taking photographs or filming this session, especially flash photography, unless otherwise directed by the presenters. For those of you looking for Wi-Fi, the network password is ASGCT 2022. You can download the ASGCT Events meeting app from the App Store. So we have two panels today. Let me see if I can forward here to get the meeting up. Oops. Go back here. I don't know if we were going to have the agenda up. I should have asked. So we have two sessions today, this afternoon. One, a panel talking about clinical trials. So starting with current trends in clinical trials, how a clinical trial is set up and run, what are the complexities that relate to eligibility, and then what to expect before and after, before and during a clinical trial, and then what to expect after a clinical trial. We're going to go through the presentations from a timing perspective and hold questions till the end panel discussions. So we'll give some additional instruction for how we'll handle questions before that session begins. If you are online, there is a chat function, so if you want to pull those in during the discussion, you can do that as well. So I am honored to introduce our first speaker and my co-chair for this workshop today, Dr. Kimberly Goodspeed. Dr. Goodspeed is a child neurologist and neurodevelopmental disability specialist who works in Dallas, Texas at the University of Texas Southwestern. Dr. Goodspeed became interested in rare genetic disorders as a physician in training and has leveraged that passion into a career dedicated to changing the way developmental disorders will be treated in generations to come. She's leading a number of clinical readiness programs, including studies for SLC 6A1 related disorders and SLC 13A5 deficiency disorders. Also to note, in 2021, Dr. Goodspeed was rated one of Dallas's best pediatric specialists by D Magazine, and when you meet her, you'll know why. Today, Dr. Goodspeed will kick off our session one with her talk on current trends in clinical trials and gene therapy. Dr. Goodspeed.
All right. Oh, I am almost too short to see over. So, I'm Kim Goodspeed. I'm super excited to be here today, and I'm really excited to be co-chairing this with Emily. It's been a lot of fun to pull this together, and I hope it's informative for everybody who has ventured on this with us to attend. I'm kicking us off just by going over some of the basics and taking a real bird's eye view, and then letting my co-speakers, who were really excited, are coming up behind me to dive down into the details of each one of these aspects. So a whirlwind tour through clinical trials right now. So, where are we headed? I want to level set a little bit and just talk briefly about precision therapies and clinical and translational research more broadly, give a broad overview of how clinical trials are designed, just kind of a quick setup, what are controls, those kinds of things, and then how do we prep for these clinical trials from the clinician's perspective. Here's some disclosures. I work with a couple of companies on some really exciting research projects, and I've had some consultative work as well as working with Combined Brain. So, precision therapies, what are they? I think this is turning into a little bit of a wastebasket term now, but I think of them as any kind of targeted therapy. So, of course, we're at a gene and cell therapy conference, so we're thinking mostly about that, but a supplement or a special diet could be considered a targeted or a precision therapy. Also, small molecules. We're, of course, not really focusing on those today because of the type of conference we're at. But mostly looking at gene therapy and RNA-based therapies is kind of our bread and butter in neurology, but also CAR T-cell therapies and other cell-based therapies. So What is clinical and translational research? So bird's eye view again, we just want to level set some. So we tend to start with the patient. So we have our patient in front of us in clinic. We decide they've got a particular characteristics, and then we try to figure out what's causing those characteristics. And from there, we can decide, well, can we model this? Can we show what it looks like in an animal? Can we show what it does in a dish? Can we figure out what pathways are involved? Can we identify particular targets that we might be able to treat in a particular way? And then getting back to the patient again, can we find a particular population even within that rare disease group that these patients, maybe those with missense variants or those with nonsense variants, or if they have a particular finding on one of their clinical tests, may be more amenable to the treatment than others. And then from there, we need to think about how do we design our clinical trials and prove that these things are working and that they're safe. So there's specific phases of clinical trials that we think about. They always start with a preclinical testing. So you have to prove in a preclinical model that you have something that is safe and potentially efficacious. And especially in the pediatric population, you need to prove that there is at least a prospective benefit to those patients. Traditionally, a phase 1 study is going to be a safety study in healthy participants. However, when you've got risky therapies like what we're talking about with gene therapies or cell-based therapies, we're most often going into our disease population first. So you'll often hear that people talk about phase 1-2 trials, or even phase 1-2-3 trials. Now, what's a phase 3 trial? This is the study that typically focuses on efficacy, and that's usually the registration-enabling trial, the trial that brings a drug to market and makes it available for more patients outside of the clinical trial. And then our phase 4 studies are those long-term studies looking at the long-term outcome effects, long-term safety as well as efficacy. All of this in rare disease gets a little bit muddy, so the barriers aren't quite as well defined, and it may be different for different patient populations and different treatment options. So it's important to work closely with your regulators and your clinical teams and your drug development teams to all figure out what's the best option for this particular treatment. And so here, I'm a US-based physician. I work in Texas. And so I come from the bias of thinking about how the Food and Drug Administration thinks about these things. And so this is a quick overview that's put out by the FDA, and I think it's a nice infographic. I'm not gonna belabor it too much. It really just puts into kind of a story timeline of the slide that we just looked at. So a drug's developed, it's tested in animals, and then we have an IND application. INDs are huge ordeals to take on most often. It's an investigational new drug application, and it puts everything out there for the FDA to review, from all of that preclinical testing, the safety assessments, and what's your plan going forward into humans. And then you go through your phases of your clinical trials, and ultimately, you end up with a drug that's out on the market and is being used to treat patients. And we continue to follow and get information on how it's being used and how it's working. So that's the broad overview of what are the different phases of clinical trials, but there's a lot of different ways to design those clinical trials. Traditionally, we would have a placebo-controlled, double-blinded, randomized clinical trial. That was kind of the gold standard for a very long time, but there's a number of things that make it harder, especially in rare disease, and especially with treatments like gene transfer therapy that can only be given once. So in this traditional model, you identify a group of patients, recruit your patients, you randomly assign them to either get the experimental treatment or to be in the control group and get a placebo or a standard of care or some other control measure. And then you follow people over time and you measure them the same way, and you decide at the end of it, was it safe and was it effective? But this means that half of your patients aren't getting the treatment. And in the rare disease world, that's a hard pill to swallow, no pun intended, for lots of families that are wanting help and now and don't necessarily want to wait. So, rare disease research can be messy. We know that patients are out there. They're getting lots of different treatments in lots of different centers. There's a lack of clinical care guidelines, and you don't always know if the outcome you're seeing in the clinical trial would have just happened anyway, or if it's truly an effect of that drug. And then how do we control that environment, and how do you balance the risks and benefits and talk through all these issues with our families? So all of this is going to be touched on a bit throughout our sessions today. But one important piece is the control group. So I keep saying, how do you know it worked? How do you know it's safe? In the traditional model, we have a group that's telling us that, the placebo group who's not getting treated, and we can see are there differences between them. But in the event that we aren't using a placebo control group, we're often going to be looking to historical controls or looking to that historical control within the particular patient or participant for their baseline measures. That means that those historical controls are really important, and we're going to talk a little bit more about that here at the end. There's a few different ways to design these trials too. I'm going to highlight a couple here that I think are pertinent to the types of therapies that we're talking about, starting at the end of one clinical trial design. Again, this is going to get touched on a bit more in session 2, but in this design, you have a single study subject, one patient, who is going to be your trial participant. You do some run-in period baseline measures with that participant to decide how do they look now before you've treated them. You give them the experimental therapy, and then you measure those same outcomes again and compare the post-treatment to the pre-treatment assessments. Now, if you're able to have a washout period, maybe it's an RNA-based therapy and you might be able to wait a period of time and dose again, you can do that and measure again. In the case of a gene therapy trial, though, this is a one and done. So it would be a single time point. So there, even within these adaptive or alternative designs, there's still some things that you need to consider about your particular treatment. The randomized placebo phase is another interesting way to go about this. And it can be designed, again, in a few different ways. But in general, you identify your subjects, you randomize them to either get treated now or treated later, and you follow everybody over time. Now, you don't necessarily have to give a placebo, but you could randomize the time at which you give a treatment and blind some of your assessors so that they don't know. Maybe your physical therapist doesn't know when a patient got treated. And so that removes a little bit of their bias and if they're hopeful that treatment is working. And this can be a little bit more palatable to a rare disease population where they know they're going to get treated, they're going to receive the experimental intervention, but we have a little bit more rigor added into the design. Now, all of these are hinging on needing to know what patients look like without treatment. And so observational studies are super important for that. Now, they can start with easy things like a disease registry or retrospective study. Those are generally fairly cheap to do, fairly easy to do besides the burden of filling out 1 million questionnaires for our families. But they give us a lot of great information on which you can build a really good prospective study. So retrospective, looking backwards, prospective, looking forwards. So in your prospective studies, you grab a group of patients, you follow them over time, and you do the same assessments at standardized intervals. A lot of times in clinical practice, I'm getting a test because I have a clinical concern. In the confines of an observational study, though, that bias gets removed and we see what everybody looks like at standardized time points. It can also help you identify biomarkers. Now, what are biomarkers? We'll get to that in just a couple of slides. A great example, though, of this in practice was the spinal muscular atrophy group. So they did a great natural history study that identified the time points at which you could expect patients to hit a particular clinical endpoint. In this case, it was death or mechanical ventilation or invasive respiratory support. And they used that as their comparator arm. So this red line demonstrates when a patient hit one of those undesirable endpoints. And the blue line at the top represents all of the patients in that clinical trial. And that was pretty compelling evidence that the clinical trial had a big difference. So again, endpoints are really important. Death and invasive inhalation are very clear endpoints. They would be clinical endpoints though, and sometimes we're using biomarkers. These are surrogate endpoints. So things like a blood test. Maybe you have an inborn error of metabolism and we can measure something in the blood that is elevated and then we can show it goes down with treatment. Maybe you have some electrical testing or electrophysiological testing, EEG, nerve conduction studies, brainwave tests, things like this, and we have a particular signature that we're watching and eliminating with a drug. Or maybe there's an imaging biomarker that we can follow. And then the clinical outcome measures are anything that measures how a patient feels or functions or survives. And so these are going to be things like, wrong button, performance measures. So cognitive outcome measures. How are they performing on an IQ test? How are they performing on a motor assessment? Quality of life measures or proxy report or parent report or patient-reported measures. How does the patient feel when you ask them what their symptoms feel like? Or physician rating scales. How does the physician see the patient changing over time? And all of these get lumped together to help us decide, is a particular new treatment helpful? And the decision to use any number of these is coming out of those observational studies. So, what's new in gene and cell therapy here in our last minute? Quick whirlwind just to highlight some of the accomplishments that have happened over the last few years. We have 19 gene therapies that are currently approved. That's super exciting. 17 RNA-based therapies and 56 non-genetically modified cell therapies. What's in the pipeline, though, I think is even more exciting. Over 3,500 therapies are currently in development, almost 2,000 of which are gene therapies, and just over 800 are non-genetically modified cell therapies. And who's getting treated? What are we going after? And over again, it's anti-cancer drugs and rare disease at the top of the list. Rare disease is a big catchment area, but I think it's a really exciting thing to see that rare disease has risen to the top and it's found its niche and people are focusing on it. And when you look at RNA therapies, it's a similar story, RNA and cancer. And if we drill down a little bit in the RNA therapies and what's actually being treated, as a neurologist, I have a little bias here, but I remember entering this field as a time when there was nothing available, and all of these are now in development for neurology. So really cool time to be a neurologist. So what's coming up next? Emily gave you a quick overview of our agenda for today. We've got some really great speakers coming up behind me. Again, we're gonna hold questions till the end and do a panel discussion. And then our session 2, I hope you'll stick around, and it gets a little bit more patient-focused and story-focused, and what does this all mean and what's it for? And I always end with this because rare disease needs everybody yelling the same thing in order for everybody to hear it. So we're about communities here. And with that, we'll move on.
Thank you. Thank you, Dr. Goodspeed. Our next speaker is Megan Waldrop. Dr. Megan Waldrop is a pediatric neurologist at Nationwide Children's Hospital and an assistant professor of pediatrics and neurology at the Ohio State University College of Medicine. Dr. Waldrop completed her pediatrics and child neurology training at the Children's Hospital of Orange County and University of California, Irvine School of Medicine. She then pursued a neuromuscular medicine fellowship at The Ohio State University, Nationwide Children's Hospital, followed by a neuromuscular genetics therapeutics fellowship at Nationwide Children's Hospital. Her clinical and research interests include clinical translation of genetic therapies and equitable healthcare delivery in neuromuscular diseases. Please welcome Megan, Dr. Waldrop, to talk about how a clinical trial is set up and run.
Good afternoon, everyone, and thanks to the committee for inviting me to speak. I just want to let you know that this is based on my experience at Nationwide Children's Hospital, and there may be a little bit of variability between sites, but this is just designed to be a general overview to give you a little more information about how we kind of set up and run our clinical trials. And I was thinking about this question when I started this talk. So let's say that a certain product was approved for clinical trial by the Food and Drug Administration today. So we're thinking this, parents and families are thinking this, when can we dose? When is the trial going to start? And we would all love it to be the next day. But unfortunately, because we are really focusing on patient safety and good, strong trial data, there's a little bit more that we have to do before we can dose that first patient. And I'm also doing a few assumptions giving this talk today. So we're assuming that all preclinical studies have been completed. So we know that it's safe in animal models. We know that it's effective in animal models. We've picked an appropriate dose. And we have additional safety data in a large animal model that the FDA tends to like, the non-human primate, because it's most like humans. And I'm also assuming that the IND, or the investigational new drug application, has been submitted or approved. And this is only a single site study. And it's a phase 1/2A study, which is really relevant in kind of the rare gene therapy space. So before the IND can be submitted, though, we've already done a lot of work. When the researcher is pretty close to thinking about approaching an MD for designing the clinical trial, that's when we start preparing. So who are we going to include in this trial? What is the patient population going to be? What age groups are we going to include? How are we going to assess the safety? What are we going to use for our outcomes? Do we want to include additional efficacy measures that we're not certain could show efficacy, but might? We're not sure. And then who are we going to include and who are we going to exclude? That all has to be done before the IND is submitted, along with the consent form. And I bring up the consent form because we want to think about consent translation, which takes a long time. And so if you can start thinking about much earlier, you can speed things up a little bit. And so I am going to talk about moving forward everything from submission of the IND, but in reality, there's some things you could do sooner and kind of in parallel to the last stages of the IND prep. First being the Data Safety and Monitoring Board, establishing that. So it's a committee set up of local or national or maybe even international experts in your field that are supposed to be an objective third party to really review the data integrity and safety of the trial. These are usually very, very busy people and usually hard to get to give up some additional time, and then additionally to get everyone coordinated to meet at the same time. So if you start thinking about this early, it's another space where maybe you could expedite a little bit. And then thinking about, you've already established your protocol and you know what you're assessing for safety. So can all of those labs be done locally at your facility, right? This is a single site study. If not, if you have a send-out lab, does your hospital already have a relationship with that send-out lab? If not, you need to troubleshoot that because we always run into problems. And you need to go down to your laboratory and talk to them and expedite the process and do a run-through if you can. Pharmacy and drug procedure setup is another big area. So usually there's an institutional drug pharmacy kind of subset of the hospital pharmacy department, and they are staffed separately. And so not necessarily everyone in the pharmacy department can cover the clinical trial drugs. So you need to make sure that there's adequate staff. You need to pre-plan and set up and talk about how often you're going to be dosing patients, how often you're going to need them, what days of the week. Make sure they know how to prepare the drug. How many people there should know how to prepare the drug? What if somebody's out sick? How long is the drug stable? The pharmacy's made the drug. How does it actually get to the patient? Does the clinical coordinator take it? Does somebody from pharmacy? Lots of little tiny details that you could start thinking about as soon as possible. And then also just biosafety training for anyone in the pharmacy is additionally something to get done as soon as you can. Then the outcome and the efficacy measures. So phase 1/2A studies predominantly focus on safety, but we always hope that we can get some information about efficacy as well. And so you're going to have outcome measures that might be related to some blood work, that might be related to other testing, or it might be related to functional outcomes. In my area of expertise, that's physical therapy assessments. And so you need to know that the physical evaluators that you have on staff first know how to do what you want to assess, and then additionally, are they inter-rater and intra-rater reliable? Do they have all the equipment they need for the testing? Do you have the space? So some of us have very, very busy clinic days, and so you have to think about, can you do your trial patients on a clinic day, or does it have to be different than a clinical day? So additional logistics to sort out. And then the big holdup, probably the one that we have the least flexibility on, is the Institutional Review Board. So this is something that every hospital has that participates in research and human subjects research, and it's there to really ensure the safety and ethical research of human participants. And it's made up of various experts in the hospital, and also we have community experts on the board as well. There are a lot of applications that they have to review, and so typically, actually 100% in my experience, they've never been willing to pre-review something before it's been approved from the FDA. And then they usually take about 30 days, sometimes longer, to review the application. So the big holdup really is once you get the FDA approval, it's going to be at least 30 days before you can likely dose that first subject. So you have a little bit of a timeline, but things always take a little bit longer than expected. And in some scenarios, they may be short-staffed or may require a little bit longer time. There can be an additional couple of weeks. Another big thing is data integrity. So we can't just write our little notes, have every different investigator on the study do it their own way, because we need to have very strong, rigorous data to submit to the FDA to assess if this drug can go into further study, additional phases, or, best case scenario, be a registration trial and get approved after that. So we have to establish consistent, uniform source documents for everything that's being done in the study. And then we also develop an electronic data capture system. Sometimes that's done at the local institution. Other times it's contracted out to another company. Anytime you contract something out to another company, there's more time and coordinating that takes place. And then you also need to set up long-term data monitoring for your study. And again, that can be done internally or externally. And there's some political aspects, but also conflict aspects to think about that. But again, you need to have all of this set up to make the data as rigorous as possible to ensure that we're keeping the patient safe and we're getting really good data. If we're asking children and families to do this big ask for us, typically it's first in human. We really need to ensure that we're doing it as to the best of our ability. And then typically when you're getting pretty close to starting, you have a site feasibility meeting, and that's usually involving all of the key players in the hospital or the site that are going to be helping you run the trial. So there will be representatives from pharmacy, you're going to have your research coordinator there, you're going to have clinical evaluators there, really everyone there that's doing the work of the study for you, because this is not an independent task of just me. There's a huge team that really helps us get it done. You also finalize things like budget, and then if there's any sponsorship from a company, we kind of help nail down all the final details of that too, to make sure that we have the funding all ready to go so we can dose that first patient. I'm not going to touch too much on this because the subsequent talk is going to talk about this a lot. I just want to say that it's not so simple as we've picked who the first candidate is going to be. Now we can just get everything scheduled. So typically there's a fair amount of screening procedures to determine if someone is even eligible. And that may be done in various parts of the hospital, done by various providers. And so the clinical research coordinator has a bit of a task, a little bit of a puzzle to fit together to get everything scheduled that needs to be done as quickly as possible, but also trying to minimize the inconvenience to the family with multiple trips back to the hospital. And we schedule all of that once we know which patient we're going to offer the spot to. But again, that's the caveat that this is only if they sign the consent form and agree to proceed. If the consent form is not signed on that date, if the family needs more time to think about it, then we'd have to kind of shift and push our schedule a little bit more. And then throughout the study, there's continued data monitoring and review. So the DSMB that I talked about in the beginning, they have to meet 30 days after the first patient has been dosed. And this is really to ensure the safety of the patient and also that the clinical team is running the trial up to snuff. So they review all of the data and they have the opportunity to ask the clinical team any questions, make suggestions perhaps if, oh, in hindsight, maybe we really should add this on as a safety lab, or maybe in hindsight we should space timing between this. And so that's another kind of key time-limiting factor to dosing additional patients, right? It's kind of hard sometimes to get everyone on that committee to meet exactly right at day 30. And we do our darndest, and we're usually pretty good to get it within a couple of days. But we're not allowed to move on to additional patients until the committee has had time to review all of the data and make their decision and recommendations. And then they continue to meet every 6 months throughout the study to really focus on the integrity of the data, but first and foremost, really the safety of the patients. And then we have continued clinical trial monitoring as well. And again, just making sure that the data is up to snuff and we're doing everything as rigorous as we can. And that is all, and I'll take questions at the end. Thank you.
Thank you, Dr. Waldrop. I'm now pleased to introduce Dr. Heather Lau. Dr. Lau is a board-certified child neurologist and currently an executive director in global clinical development at Ultragenyx. Prior to joining industry, for the past 10 years, Dr. Lau was the director of the Lysosomal Storage Disorders Program and associate director of neurogenetics at NYU School of Medicine, where she managed a diverse cohort of patients with rare neurogenic diseases. She was principal investigator for over 20 clinical trials studying small molecules, protein, and gene therapies for a variety of disorders. She is now adjunct associate professor at Yale University and partnering with Dr. Pramod Mistry to continue her academic endeavors on a part-time basis, as well as continues to serve on advisory boards for patient organizations. Today, Dr. Lau is going to talk about complexities of eligibility. Why do we have to exclude patients from gene therapy trials?
Thank you so much. Thank you for having me here today. So let me just forward. Here are my disclosures, which are extensive. I've been conducting rare disease clinical research for over 10 years, and I've partnered with a variety of sponsors over these years, in both as a PI and as a consultant, and also an advisory board member to patient organizations. But most importantly, I was a caregiver, physician to a large cohort of patients with lysosomal disorders. So today is International MPS Awareness Day, and I'd like to just remember this patient with permission, Alex. And I'm going to use Alex as an example as we go through the complexities of eligibility. Alex had MPS 6, and she passed away about 2 years ago. She also had a second diagnosis of a rare genetic disease. So why do we limit access to a trial? A study is designed to demonstrate whether an intervention is safe and efficacious in a specific time period for a specific disease. The sponsors of the trial must generate enough evidence that their intervention will support a regulatory decision in the population most likely to benefit from the treatment. And how do we do this? We do this by defining that study population. So the eligibility criteria are limiting the variability within that study population to control for these confounding variables, to maximize the benefit or the probability of detecting that this intervention is working if one exists. So how are these eligibility criteria determined? Researchers generate this list to specify that population. So what are the symptoms of the disease that can be observed and measured within that time frame? And this list is known as the inclusion and exclusion criteria. The eligibility criteria should generally be sufficiently narrow to minimize that noise so that we don't mask a finding that a treatment is working. But it should be sufficiently broad so that it's generalizable to the population outside of the study. It needs to be feasible, especially important in rare disease. We can't have the most perfect study designed. We can't have the most perfect study subject. These are humans, and clinical research in rare disease is messy. So we need to have a feasible recruitment strategy as well. And this is all so that we can detect a difference between the study group and the control. And as we heard before, ideally these trials would have a randomized, controlled, double-blinded setup. But in rare disease, oftentimes we're relying on historical controls, or patient is their own control. Regardless, we need to see a difference between before and after. So just some definitions. What are inclusion criteria? They're the characteristics that are specified that a patient has to have in order to gain entry. Right, so we don't have the ability just to enroll anyone who wants to be enrolled. We have strict criteria to allow us to define this population to study within a short time frame, right? These trials cannot go on for years and years, not at least in the registrational trial. We heard about phase 1, phase 2, phase 3, but when we're trying to get to that important outcome, whether or not we see a treatment effect, we have to identify this population. And not only do they have to hold the disease, such as in in gene therapy, we need to verify the genetic mutation that's conferring the diagnosis, but they might have to have a minimum threshold of disease severity. We can't always enroll asymptomatic, and occasionally we can enroll pre-symptomatic, but that's not necessarily the rule, but the exception. So what are exclusion criteria? These are the criteria that disqualify the participants from a study. That might include later stages of disease. But other factors include comorbid medical conditions or other treatments or medications or factors that could mask the effects of the intervention. For example, a second genetic condition. So in my patient Alex, she had MPS 6. Initially, she was misdiagnosed. So we had to confirm diagnosis because she looked like she was an MPS 1 or 2 because of her cognitive impairment. But in fact, on further investigation, she had a second diagnosis that led to the cognitive impairment and the optic atrophy. If she was to be enrolled in a study that looked at, say, outcome measures of cognition or even in vision, it would mask the outcome of the primary intervention because she had a second diagnosis. Use of experimental or off-label medications. This is difficult. If you see a treatment benefit, is it because of the medication you're testing right now, or is it the second medication that the patient is on? Sometimes you'll see requirements to maintain your medication regimen. This occurs in epilepsy trials often, where we don't want the patients to come off their medications, but they have to maintain stability in order to understand whether that new medication or that gene therapy is having a positive effect. So how do we define, or what are the factors going into defining the eligibility criteria? So just like in medicine, where the patient is at the center of the circle, here in drug development and in looking at criteria, the person again is at the center. And we're looking at medical conditions, we're looking at the intervention that we're studying, whether it's a drug or device or procedure. But we'll also look at other factors, demographics, what kind of examinations are required to go into the screening of the person, gender-specific conditions, lactation, pregnancy, or willingness, or to not get pregnant during a trial. All of these go into creating the list of inclusion and exclusion criteria. And the other category, just to highlight, there's life expectancy as well. Is this a fatal disease? Is it rapidly progressive? That might have different criteria or looser criteria to allow patients into the study. But if it's more attenuated, you might see stricter criteria to mitigate risk for that patient. So examples. So looking on clinicaltrials.gov, they actually list the inclusion and exclusion criteria. And this is very empowering for patients and their caregivers to be able to see what's happening in their disease state, what research is ongoing. And that list of criteria is helpful, but please don't make a decision based on reading it by yourself. Please reach out to your doctor. the doctor or the principal investigator can walk you through these inclusion-exclusion criteria. So for today, I just wanted to give you a few examples. This is taken from a real study. I didn't disclose which one, but it is a gene therapy trial. And you'll see some exclusion criteria that include current participation in another trial, which I explained, would confound the results if there was a positive treatment effect. But in gene therapy, there's another issue. It's irreversible at this time. It's a one-time injection. So we have to exclude patients that have been in previous gene therapy trials, or in bone marrow transplantation, we use that oftentimes for leukodystrophies and other neurodegenerative diseases. That would lead to an irreversible change in that patient and might preclude them from a gene therapy trial. Here we also list unrelated medical conditions, like my patient who had 2 genetic conditions. Previous use of any kind of con-med or abnormality that would preclude puncture, lumbar puncture, or ICM injection on MRI, or undergoing MRI. So if there's something, low platelets, if there's an abnormality in the structures that would this is not related to the primary intervention, but in either delivering the intervention or assessing whether or not the patient is stable. And then here are the abnormalities of the labs. Now, these could be transient. That's why I say don't necessarily look at the list of criteria and automatically assume that you're not eligible. Work with your doctor and talk to them. How do I get myself in top shape for a clinical trial? How do I make sure my patient, or my child, or myself are healthy enough to withstand the rigors of a trial? Okay. Primum non nocere, first do no harm. This is a concept central to all of us in medicine, and it can be applied here in clinical trials and clinical research, most importantly, but also during the screening process. So let's go step by step. The way I think about it is that at the time of consent, we're going to discuss not only the risks and the benefits of the intervention, but we'll also discuss the risks and the benefits of the screening assessments and all of the diagnostic assessments, because they are not necessarily without risk. So the questions that you should ask as a parent or for yourself: what type of assessments and procedures must I undergo in order to establish my eligibility? Is there risk associated with this screening process? And then we have to weigh the risk and the benefit of simply just being in the screening process. You may not ultimately be eligible, but are you willing to undergo invasive procedures to establish that eligibility? How can we design the study? How can the investigator, the sponsor, design the study to minimize this risk up front to the subject? And so I gave an example on the right, escalating the screening procedures. Oftentimes, at time of consent, we might obtain initial screening information, confirm the disease based on your genetic report, understand your severity of disease, taking on a thorough history, undergoing a thorough physical exam, and then we might escalate. Okay, we've confirmed your genetic diagnosis and some basic information. Now you pass on to the next phase of criteria. We assess your health status through more invasive means, possibly a blood draw. And then again, we pause and we verify eligibility and we move forward. As we get closer to dosing and the potential for enrolling into the study, you might undergo more invasive procedures. For example, in pediatric patients, anytime we have to sedate, there's a risk. And so we would want to hold off on those procedures until later in the process to make sure that that child's eligible, and then undergo the sedative procedure, the lumbar puncture, the MRI. And then we can verify eligibility and prepare for enrollment. So ethical considerations, vulnerable populations. Historically, research studies have consistently excluded certain groups of patients. A common one is patients with organ dysfunction or multiple chronic medical conditions. We exclude them because we don't want to do harm with our therapy. We know the general risks. We're going to find out more risks as we enter phase 1, phase 2, and we don't want to subject a vulnerable patient. But that leads to exclusion of, in certain cases, older adults because they have comorbid medical conditions, they have polypharmacy. But it can lead to, it can also exclude children who have other medical problems. Studies have also excluded patients, pediatric patients, pregnant and lactating women, due to concerns of the impact on that fetus, the infant or child, and what are the long-term effects. However, what happens here is that it leads to an evidence gap. We don't know what is really safe and efficacious in the real world. So our patients might have access to it after it's approved, but we've never studied it. How do we know what dose to prescribe our patients if we didn't understand it in the research trials? So there are ways to design trials or companion studies to look at these vulnerable populations, and also looking at real-world evidence. So for example, you'll see dose adjustments and renal impairment, and then we get a better understanding of the pharmacokinetics of a drug. This is harder in gene therapy where it's one time only. But again, we might take a look at a subgroup. So you might be excluded from the primary trial, but there might be opportunities to include you in other phases of development. In pediatric patients or in pregnant women, oftentimes we look in post-marketing data. So we follow patients forward, patients accidentally get pregnant, and then we understand better about the safety of that drug. So in 2018, in an FDA workshop, they actually highlighted this issue, and there's initiatives to increase the inclusion of subgroups in clinical trials to ensure that women and older adults and others are investigated or have opportunities to have access to these trials, and to ensure that all eligibility criteria are scientifically justified. And these are undertaken both through the FDA and through the NIH. And they encourage all of us to make sure that our inclusion and exclusion criteria are scientifically justified. So how can a researcher communicate to the patient community about eligibility for a trial? Well, I first told you about it. The list of eligibility criteria are on clinicaltrials.gov. But as a physician or as the PI, we need to explain to you the rationale for why we created that criteria. What is the background to why we're excluding certain patients at this time? We need to manage expectations related to the potential for screen failure as well. So you're undergoing these assessments. What is the likelihood that I will go on to be able to have access to this drug or this therapy? We need to know the types of assessments and the risks associated with it. And then we have to be transparent in why you failed. Why did you not pass on to eligibility? Why did we exclude you? Could the patient be eligible for future studies? This is critical. If we know at the time, we should share it with you. We may not know, especially in early phases where we're not sure if it's efficacious. We may not want to have these vulnerable populations exposed to a therapy where we don't know if it could be potentially beneficial. And could we have access to this therapy in other ways? And in certain ways, we can use innovative designs or alternative studies, companion studies, to reach these patients that are initially excluded from our trials. So we have to think about the impact, not just participation in the clinical trial itself, but this screening period specifically. It requires time, financial resources that are unreimbursed, but the psychosocial impact should be at the forefront of the entire treating team, of the team that's involved in research. The subject needs to understand that they are being asked to participate. They can refuse to participate, but they also don't have a right to access. They could be excluded. We have to manage this expectation, this perception that they're being excluded from these studies. It's for safety. It's also to give us the best chance that this drug may or may not be working in a short time frame. And hopefully, we would be able to extend this to all patients with this disease later. But initially, we have to start with a narrow group. So there's lots of factors that are related to patient participation in trials, and they looked at this in this paper here. Barriers, there's many barriers. There's fear, there's distrust, there's treatment preference, perceived stigma, practical difficulties, and concerns about the trial design. You heard about randomization and placebo, which could be very scary for a parent, especially if the child is having a either rapidly progressive or fatal disorder, that the idea that they might be randomized. Now that's reduced in the rare disease world, but still it could be on the minds of our caregivers, even if it's delayed, even if it's a roll-in placebo period. And the incentives for our patients are potential for personal benefit, obviously, altruism and trust. Okay. So when we think about eligibility criteria, and we start all the way at the beginning with informed consent, it is up to the PI and the sponsor to make sure that we fully describe all that we expect of our patients if they participate even in screening for a trial. Why do we have narrow eligibility criteria? To generate a homogeneous population, and hopefully we can see that this therapy, there might be a signal that this is working. And then we hope to use broader inclusion criteria to allow others involved into other studies to get a better sense of the real-world effect on the population. And if you combine the data from the pivotal trials and these other trials to give a comprehensive review of the safety and the efficacy, it'll allow us to better serve our patient population. But it is a challenge for sponsors to weigh this, the ideal study versus the real-world use of the drug. It's important for all of us in research to take into these considerations the ethical impact of exclusion criteria, to execute initiatives to ensure inclusivity of our traditionally vulnerable patients, as well as understand and manage the psychosocial impact of this whole process for our patients and caregivers. It's important for the researchers to properly inform the patients of the risks and benefits of all aspects of participating in a trial, including the screening period. Thank you.
Thank you, Dr. Lau. Just as a reminder, we're holding questions till the end for the panel. But for those of you that are joining us online, there is a chat box where you can start placing your questions there. And then if you see questions that pop up, you can also vote on those to raise the questions you feel that are most important. Our next speaker is Dr. Nicolas Abreu. He's an assistant professor of neurology, associate director in the Division of Neurogenetics, and director of lysosomal storage disease program at NYU Langone Health. Dr. Abreu began his interest early in neurodevelopmental disorders as a peer mentor in high school who has a range with children and classmates who had a range of physical and developmental disabilities, including cerebral palsy, autism, and intellectual disability. This led him to pursue a career in child neurology and neurodevelopmental disabilities. During his residency, he became interested in identifying a genetic cause for a neurological condition so that he could provide patients and their families with an answer to the long-asked question, why did this happen? At NYU, he evaluates and treats people of all ages who have complex neurological disorders, such as developmental delay, seizures, or difficulties with movement. He also manages lysosomal storage disorders. Thank you, Dr. Abreu.
Hello. So today I'm going to be approaching this conversation as if talking to individuals affected with medical conditions that may be amenable to gene therapy or cellular therapies, and given my background, specifically rare neurogenetic conditions. And we'll focus on 4 key areas, going from considering a clinical trial, which we've heard a lot about, to the process prior to gene transfer, to the gene transfer itself, and the immediate post-gene transfer period. So once an individual or family has a loved one diagnosed with a rare genetic condition, oftentimes there's a search for answers. One, not only why did this happen, but crucially, what can we do about it? How can we move forward? And oftentimes there are conversations with multiple healthcare providers trying to get answers, as well as looking online and social media to try to figure out what the next steps might be. Critically, your healthcare professional should help you provide some direction in terms of clinical research opportunities. It's not always the case that any given healthcare provider is up to date with gene therapy clinical trials, as certainly this is a niche within the greater world of medicine. However, it's certainly a growing niche, and one that we hope with symposia like these that we can expand our reach. Working with your healthcare providers, oftentimes the first step is reviewing one website called clinicaltrials.gov, which we've already heard. This is a database by which the US Food and Drug Administration, as of 2007, has required interventional clinical trials, especially those that require biological agents like gene therapy, to be registered. It is not particularly user-friendly, and so for families that are looking at clinicaltrials.gov, as was mentioned in the previous talk, to go back to your healthcare provider after reading any information that you might glean from clinicaltrials.gov to see what the next steps may be would be helpful. But importantly, clinicaltrials.gov lists study sites as well as contact information of potential sites in order to get connected. In order to improve the user-friendliness of finding a clinical trial, the American Society for Gene Cell Therapy has created the Clinical Trials Finder specifically around genetic and cellular therapies, and so may be another resource. Finally, patient advocacy groups and disease-specific foundations have a long history of compiling information for rare disease individuals to find particular avenues for clinical research and continue to be an amazing support for newly diagnosed families about getting connected. Let's say an individual or family has found a potential clinical trial of interest. Where do you go from there? Really, for any individual or family, it's defining your why. Why go through the labor of an experiment or a clinical trial as opposed to standard of care? Most often, this is because there's an unmet need within the current medical care that is trying to be addressed by this given therapy. It is critical to understand that any clinical trial, even a gene therapy clinical trial, while we hope for benefit, really should not be seen as a cure. However, other reasons to participate could be, as mentioned previously, things like trying to benefit the greater scientific community or other individuals who may be not immediately available to benefit from a therapy but could benefit from the future. So thinking about the why is critical. This why can change over time, so it's not just a single point in time that you decide, I'm going to move forward. There can be reasons to take a pause in the search and reinvigorate the process later, as well as it can vary between family members. And some people can have very strong feelings about whether to or not to pursue a gene therapy clinical trial, and it's important both within a given family to make sure all voices are heard, including children as well as siblings, in addition to working with your healthcare providers to make sure that when embarking on the process that everyone is respected. Secondly, what gene therapy clinical trial is currently available? When a clinical trial is posted on clinicaltrials.gov, it doesn't necessarily it's not immediately obvious whether or not there is even an intervention. So families may see a natural history study, or you may see something that only describes something called biomarkers, which may not actually provide a specific treatment, but is still critically important to advancing the field. So understanding whether or not there is even a therapy offered through a given clinical trial is important to determine early on in the process. Within a given gene therapy clinical trial, how is it being administered? What is the particular dose? And some other considerations about has it ever been studied before? Is it a first in human? These kinds of things, working with your healthcare providers can be helpful in the process of determining whether or not to pursue enrollment. who is involved in a clinical trial can oftentimes seem opaque or mysterious. The two key groups or stakeholders in terms of running a clinical trial includes the sponsor, or the primary organization funding and responsible for a given clinical trial, as well as the investigator or principal investigator that is overseeing on the ground the execution of a given clinical trial. This can be split up on the PI or principal investigator level at multiple sites, or potentially a single site. And within a given site, there can be many individuals who we've heard from, nurses to physical therapists to proceduralists or surgeons if needed, and various different roles. And of course, one of the most critical person who families will be communicating closely with is the clinical research coordinator. Often, it is very exciting to see a clinical trial, especially a clinical trial that may be open for enrollment, but one of the important issues to determine whether or not a family wants to participate is where are they currently enrolling patients. Sometimes it can be a single center across the world. Other times it can be multiple, and centers can come on or off over time, and families, while it can be exciting to see a center close to a given residents to come, oftentimes there may be a requirement to travel to another state to participate. And oftentimes travel to another state may be for multiple visits or potentially for for multiple months. And when is a clinical trial enrolling is an incredibly difficult question that many families face. In clinicaltrials.gov, there's oftentimes, or they list which studies are currently open versus terminated, and if they are open, if they are actively recruiting, or if they are no longer accepting new patients. This is only the tip of the iceberg when it comes in terms of whether or not an individual would be considered for participation in a clinical trial. And so ultimately, it's important to talk with the investigator team directly rather than waiting for something to appear on clinicaltrials.gov. And so certainly better to work with your healthcare team as well as with the investigators directly rather than waiting in the sidelines for something to appear online. Assuming that you've found a clinical trial that you're interested in participating in, there are a series of steps even prior to gene transfer that we've heard of a little bit thus far. And that ranges from pre-screening, so prior to completion of informed consent, where oftentimes there's routine clinical care being provided, but where there's information provided to families or affected individuals about what the clinical trial may involve. This can come in the form of a telehealth visit, a telephone call, or piggybacked off of a routine clinical visit, and is at the beginning of the process of understanding what a gene therapy clinical trial may involve. It sometimes may involve testing for things like AAV antibodies or a pre-existing immune response to gene therapy related to adeno-associated virus, but not always. And importantly, all that is done prior to informed consent should not be considered as research since consent has not yet been obtained. Informed consent is a process. It is not just a document, but very often the focus when enrolling an individual into a gene therapy clinical trial is in the informed consent process comes down to a form or an agreement, not a contract, between the individual and or legal guardian who can sign on behalf of the individual, as well as the investigators for permission to participate in research. Once the informed consent form is signed, then screening procedures, including any imaging, laboratories, or assessments that need to be done purely for research purposes can be conducted, and reviewing of inclusion and exclusion criteria can be made. But the idea is for pre-screening before informed consent is obtained to make sure that a trial moves quickly, that there can be some narrowing down of who then proceeds on for the clinical trial. And sometimes screening and a baseline visit, meaning a repeat evaluation of current functioning, feeling, and behavior prior to gene transfer is done. That's a critical point to understanding what the individual is doing or behaving immediately prior to gene transfer. Again, sometimes screening and baseline can be combined together. Again, despite the fact that informed consent can often be boiled down to a form, it is an evolving process that requires ongoing dialogue between the principal investigator and study staff, as well as the family. And so questions from both sides should be asked and answered throughout prior to gene transfer, as well as continuing on forward. Sometimes, or oftentimes, informed consent forms need to be updated and revised as new information or changes to the study protocol are made. And so there can be a process called reconsenting, where families have to sign updated consent to any changes to procedures. To highlight some more details about informed consent, it is critical to understand that for clinical trials, this is an experiment, and ultimately not, even though we hope for benefit, we by no means can guarantee it. And understanding appropriate risks and benefits prior to proceeding with gene therapy prior to proceeding with gene transfer is critical. And so families should be totally open and asking their investigators about what they foresee as potential risks and potential benefits. And sometimes there can be some difficulties in making sure that families' expectations are met with where we currently understand as the science. The whole reason why this needs to be done in the first place is that we don't fundamentally know what the outcomes will be, even though we are hopeful for positive outcomes. And so maintaining equipoise is critical of investigators. In addition, families should be aware that informed consent or providing informed consent is optional, and that even if they're working with a treating provider, enrolling in a clinical trial is voluntary and not something that they feel that they have to do. But we do hope that it will advance science, as well as potentially have and potentially serve them, even if it is not immediately obvious. And finally, in the informed consent form, there should be some mention of privacy in terms of if you're sharing specimens, who is it shared with, and for how long. Finally, assuming that an individual proceeds through the informed consent and enrolls in a gene transfer clinical trial, the gene transfer day, or day 0, is typically thought of as the trial in many people's eyes, that the next step prior to enrollment is just to get to this day. Unfortunately, it's a lot more complicated than just getting a gene therapy. And so this is a protocol, at least the pre-transfer and immediate post-transfer protocol developed from the spinal muscular atrophy gene therapy clinical trial led by Dr. Mendel and colleagues from Nationwide Children's Hospital. And here you can see the visits in the middle column, or middle row, outlined 1 to all the way to monthly to every 6 months. And on the bottom row, you can see how it relates to the days in the study, all the way from at least 30 days prior to transfer, all the way to day 0, or the gene transfer day, and then the subsequent follow-up visits. Each visit is not necessarily just a clinic visit, meaning a 30-minute checkup. It is much more detailed than that. And so it can require extensive time on behalf of families or participants in order to complete any one of these visits. Within the vector infusion or the triangle period, you can see here for this particular trial, the gene transfer was done in the hospital and required a multi-hospital day stay for the gene transfer, even though now it is conducted on a commercial level as an outpatient. For this particular study, it was done in the hospital, needing a continuous time away from home, away from loved ones, to be able to complete the study procedures. During these days -1, 0, 1, and 2, there's many things going on apart from just a gene transfer. Individuals may receive medications like steroids or other immunosuppressants for other kinds of clinical trials. Vital signs may need to be taken on a very frequent basis, especially around the time of transfer. Laboratories, whether it's blood, urine, or spinal fluid, may need to be collected depending on the study. Exams need to be done on a regular basis, and other kinds of tests of functioning, as Dr. Goodspeed mentioned in her talk, are critical in terms of both assuring safety as well as potential efficacy, especially in later visits. And questionnaires can be oftentimes as short as a few questions to multi-page questionnaires throughout the either hospitalization or the post-transfer follow-up. And this is just one example. This is not including many other types of gene therapy clinical trials, which may require many more visits or longer periods of time in the hospital. The takeaway from this is that a gene transfer clinical trial, at least in 2022, according to my understanding, is a long and laborious process that requires a lot from families or affected individuals. And so it becomes a partnership with investigators that even though an informed consent form is not binding, it really requires a huge dedication or commitment on behalf of families or affected individuals to come to all of these visits, undergo multiple procedures or assessments, and to be physically close to a study site for as short as potentially 30 days, where you're seeing that this is a weekly visits for assessments, to potentially quite a bit longer. Now, this was a wonderful quote that I found from a patient with hemophilia B treated at Children's Hospital of Philadelphia, that he said, to sum up in one line the actual gene therapy dosing procedure, "That's it? Are you kidding me?" And yes, having dosed gene therapy, it's amazing how unexciting the actual receipt of the infusion is, and that's the way it should be. But the process you can see leading up to it and then following a gene transfer is where the rubber meets the road, really. And ongoing follow-up is critical to determine the safety and potential efficacy of any given gene therapy product. And you can see that every week, especially in the first month after gene transfer, there requires close communication as well as follow-up visits with the study team. And one of the most important things to emphasize is that adverse events are things that principal investigators or study leads are really trying to pay attention for, and that can really be any change in a given health, even as minor as a cold or a fever. That's something that should be reported to the principal investigator and his or her team. So with that, we reviewed these steps just prior as well as immediately post gene transfer, and then we'll pass it off to Dr. Shieh.
Thank you, Dr. Abreu. I'm now going to introduce our final speaker of the panel, Dr. Perry Shieh. He's a professor of neurology at the David Geffen School of Medicine at UCLA. He received his MD and his PhD in neuroscience from Johns Hopkins University in Baltimore, Maryland, and completed his residency training in neurology at Stanford University Hospital and a fellowship training in clinical neurophysiology, EMG, at Brigham and Women's Hospital, excuse me. Dr. Shieh's interests include Duchenne muscular dystrophy, spinal muscular atrophy, FSHD, myotonic dystrophy, inflammatory myopathy, myasthenia gravis, electromyography, and muscle histopathology. Dr. Shieh has served as an investigator in numerous clinical trials for neuromuscular disorders. Now, pass it over to Dr. Shieh.
Thank you very much. I'm really honored to be here to tell you about what to expect after a clinical trial. Again, this is based on my observations, my experience in the clinical trials that I've conducted. I'm also addressing the patient perspective or the family's perspective in this presentation. So, what we've heard about so far is the clinical trial itself, and there are a lot of different ways that a clinical trial can be designed. We've already seen some of these, this terminology. This clinical trial can be done in a blinded fashion. There could be a placebo-controlled, and then there's a lot of monitoring, and we've heard about all the things that we are trying to measure, such as, does the drug work? You know, what are the efficacy measures? What are the outcome measures? These are the terms that we like to use when we talk about that. We also talk about safety. So what are potentially some of the side effects or adverse events that patients are seeing, are experiencing, or that we're seeing on the tests that we're conducting in our patients? And so what I'm really going to be addressing here is perhaps after that. And actually there's an extension of the clinical trial that's sometimes called the open label extension. That's actually not what I'm going to be talking about today, but I just wanted to clarify these terms. Sometimes the clinical trial, as we define it, is the shortened period of the experimental part. It might be about a year or something like that. And then after that, the patient will then enroll in something called an open label extension, which is still very much attached to the clinical trial, but it's sort of a short-term follow-up of these patients. And it's really meant to be there until the drug is approved. And because there are other patients that are enrolling in the clinical trial, and so while these other patients are being studied and while that data is being collected, everyone's data can then be monitored through an open label extension. And then after the open label extension, patients start moving into a study that we usually call the long-term follow-up. And this is oftentimes a study that's really designed to collect data after the drug has been approved. And there are a couple of different terms or different ways that this might come about. I think the most common term is a long-term follow-up. We also sometimes talk about collecting patients who have received commercial treatment. When we talk about commercial treatment, we're talking about medication after the drug's been approved. Insurance is paying for the medication at this point. And it's not technically conducted for research purposes at all. But they may receive the medication, and we may invite them to participate in a registry. And that might be also considered a form of research that might be after a clinical trial. The individual didn't participate in the clinical trial, but is participating in research where we're collecting data so that we can understand what are the long-term effects, both positive and potentially negative, of the treatment. And sometimes the registry might be attached to patients who've been in clinical trials, so that could be sort of the other term we use for long-term follow-up, so that we can understand what might be happening. And so if you participate in a gene therapy clinical trial, you will be asked to participate in the long-term follow-up study or a registry study. It is voluntary, but we do very much encourage this participation. Now, if you're somebody who receives gene therapy outside of the clinical trial, so after the gene therapy is approved, This is commercial treatment, as we sometimes call it. You may be asked to join a registry, which is somewhat in a similar vein as I just explained. And so why do we do this? Well, it's because we really do need to know what are the long-term effects of a medication, like such as gene therapy, which in theory and hopefully would be a one-time, very effective therapy. And then what is the efficacy and what are the safety effects? So what I'm going to talk about for the rest of this talk is why do we have long-term follow-up registries, and then what happens? So the why is, well, we need to understand, does it work, efficacy, and durability. And there are potentially some questions and concerns about durability in certain diseases more than others. But will gene therapy, if it's shown to be effective, will it be effective for a short period of time, a long period of time? Is it forever? And then what are the long-term safety potential effects that we might see? What are the side effects that might arise later? And things that we haven't captured through the clinical trials yet. And this is guidance for industry, a document. This most recent revision is January 2020, so just a couple of years ago, about long-term follow-up. This is issued by the FDA for administration of human gene therapy products. And in here, you can see there are some recommendations. And so you can see here, there's a whole list of things, and they've indicated specific kinds of gene therapy. So what is the vector that's being used, and what is the suggested outcome, long-term follow-up period for various different kinds of vectors that might be used. And I'll just highlight a lot of what we do in neurology is AAV-based. It's 5 years for AAV. But if you have other type of integrating vectors, such as gamma retroviral or lentivirus, which is also relevant for some of our neurological diseases, the recommendation is 15 years. And there are a lot of companies that are working in the space that really do use 15 years as sort of a guideline. And sometimes the suggested might be specific to the agent, and it might be sort of a discussion between the company and the FDA. But the FDA also says in their document, they recommend that sponsors make every effort to prevent patient loss to the follow-up to the extent feasible for completion of the long-term follow-up observations. Because this information is really important. It's really critical. It's really valuable. It's valuable for everybody. It's valuable for the science. It's valuable for our clinical care of our patients. It's valuable for the patients who have received it in the future. So we'll first talk about the efficacy and durability. How long will the treatment be effective for? And what I do want to do is just take a quick step back and talk about, give you a little bit of the components about gene therapy. We haven't seen that yet today, but so let me go through this. This is a cartoon showing you perhaps what gene therapy might be doing, and what we're really talking about here is a virus has been re-engineered to serve as a vehicle, and it's delivering a functional gene. The gene, you as the patient, that gene is not working very well or not working at all. And so this gene would come in and substitute the function of the gene that's defective, that's causing your genetic disease. And so that's what this cartoon is trying to show here. You can see this virus is carrying a different payload. It's not carrying its own DNA, it's carrying a transgene, a gene that may benefit you. So just to show you a little more explicitly, I'm just using AAV as the example because it's very commonly used for the neurology clinical studies we're doing nowadays. So this is AAV. It's a small virus, and it usually contains single-stranded DNA, and this is what it normally contains. So wild type means this is the virus that's out there that's going around infecting people. But it's not associated with any known human disease, and it has very low activity within the immune system. And different serotypes, so when we say AAV 1, 2, 3, 4 types, 7, serotype 7, serotype 8, 9, these are associated with different tissue targeting properties. So some might get into the brain a little bit better, some might get into muscle a little bit better. Those are some of the tissues that we pay a lot of attention to in neurology. And so this is just to show you the re-engineered, at least in this cartoon, form of AAV here. And so the DNA that, to go back to the previous one, the DNA that the virus contained, which helps it to copy itself and helps it to make this shell, this capsid, has been removed. And so it's now no longer containing anything that allows it to reproduce, to replicate. And so we now have these elements in here, a transgene and a promoter. And so these are oftentimes quoted as 3 important factors or 3 important things that are ingredients that are in the AAV gene therapy. I will just point out that AAV is a small virus, and so the size of the gene that it can carry is on the small side. Its payload capacity, to think about it, is less than 4.7 kilobases, so 4,700 sequences. And it also doesn't integrate, and I'll show you what that means in a second. But what that means is that the transgene could be lost with time, potentially. And just to show you what we saw in the previous slide, this is a promoter, the transgene, and the vector. And so this is housed within the vector here. And then the promoter is something that basically says when and where the transgene should be turned on, what tissues, specifically what organs this transgene is going to start expressing itself. It's going to make protein. The transgene itself is what's going to provide potentially the functional benefit. And the vector is the vehicle, so it's also important for determining how where the gene therapy is going to find itself. So this is sort of targeting. This also has some targeting factors, and then this is what's actually doing the job of gene therapy here. Now, an important thing is that integration or non-integration, and this is a little cartoon I found, which I thought it was hard to find a good cartoon that showed this, what integration is. And so what's showing here is this is the patient's own DNA, the black part, and the transgene has inserted the red part into the patient's DNA. And so that's integrating. That means it's found its way, inserted itself into the patient's DNA. What that means is that when that cell divides, when it replicates, that transgene will find itself into all the cells that come from that cell. So when the cell divides and becomes all these other different cells, it carries the transgene further. And so DNA will be copied into the tissue as it grows. And so this is what we see with lentiviral vectors, so CAR T-cell therapy or other types of lentivirus type of therapies. But with adeno-associated virus, it's a different story. The DNA does not insert itself into the patient's DNA. And what that means is when the cell replicates and becomes all these other different cells, the patient's own DNA does get copied, but the transgene does not, because it's separate and it doesn't have the factors that are necessary for it to copy itself. And so potentially what this means is there could be some dilution of the effect with time. And that's something that we get a little bit concerned about in certain diseases more than others. So this is something I just wanted to illustrate. And then, so we come back to the question of efficacy and durability. How long is it going to last for? Well, this is all theoretically right now because gene therapy is so relatively new. But one way to think about it is that lentiviral vector gene therapy, we wouldn't expect there to be a loss or a waning of the effect, the efficacy with treatment, after treatment, because it's integrated and it's being copied. Okay, that's important because we often use lentivirus in bone marrow cells, and those are dividing. For AAV gene therapy, it really depends. If you're targeting a tissue that doesn't grow at all, for instance, the central nervous system, spinal cord cells, especially spinal cord neurons, aren't known to grow they aren't known to divide, we would not expect the efficacy to disappear or wane with time. But in contrast, in tissues that might grow, at least to some extent, might divide or might have some turnover, we might potentially see the effect disappear with time. And one important question is if that happens, because this is all kind of theoretical at this point, but if it does happen, how quickly does it happen? What's the timeline of that? Are we talking about 5 years? Are we talking about 20 years? Those things, make a big difference. Those questions make a big difference. And do other medications affect efficacy? And that's also an important thing. And we can't really do clinical trials because there are so many other medicines, but we can try to make observations in what we call the real world evidence and collect trends and perhaps try to decide if there's any small signals of efficacy. And this is really information we wouldn't necessarily be able to capture in the original clinical trial. Now, what about safety? Are there side effects that arise later? Well, I go back to this figure here that I showed earlier. These integrations, they might integrate into inconvenient locations. They might cause problems. And that could turn off genes or turn on genes that you perhaps may not want to turn on. And so that's potentially a disadvantage of an integrating virus, as opposed to adeno-associated viruses, which do not promote integration. You're not as, you probably wouldn't expect those types of effects to occur. But nonetheless, that's also somewhat theoretical based on some assumptions that we have, and we don't know how rigid those assumptions are. And so it's important for us to collect this type of information for integrating therapies, where the gene may have affected nearby genes where it's integrated. This may increase the risk of certain diseases such as cancer, and this has been well documented in some of the early clinical trials where there's been integrating retroviruses used. The transgene itself, can the transgene, if expressed at really high levels, can it potentially be toxic to your tissues? We don't, presumably we haven't seen a lot of that in the clinical trials, but in the long term, we might have a better idea of whether that's something to be concerned about. And are there other effects of gene therapy, including the capsid, that might result in organ damage or tissue changes that result in problems down the line that we don't know about? There's always a lot of stuff we don't know about because we're only collecting information for a couple of years in these clinical trials. And do medications also change the efficacy of gene therapy? Again, this information is not necessarily information that we'd be able to derive from what we see in the original clinical trials. So if you have participated in a clinical trial, you will be asked to join a long-term follow-up study. And what is the burden of this? Nick just talked about how many different study visits there can be in a clinical trial. And with a gene therapy clinical trial, there really are a lot of study visits, and they can be all day. It's not a quick in and out. Sometimes you show up at 8 and you leave at 5, and there could be several days in a row, could be every few weeks in the beginning. Well, the burden in clinical trials is very demanding, but most long-term follow-up studies and registries are a lot less demanding. So I don't want to scare you into thinking that, you have to sign up for this. You certainly don't have to sign up for this, but it's also designed to really collect data in a less burdensome manner. So just to give you an idea, for most of the long-term follow-up studies I've been involved in, the visits are kind of in the long term every 6 months, and there may be some assessment of efficacy. That depends on the disease, but for us it's doing some physical therapy tasks, how strong you are. We might have you fill out some questionnaires, blood work, imaging, maybe some other tests such as looking at the heart and breathing function to make sure that there hasn't been any safety signals there. And some may offer the option of tissue biopsies. I think this is really relevant for muscle diseases because muscle is a tissue which is possibly biopsyable. Most central nervous system you would never biopsy. But with muscle, and this certainly would be optional in the long term, but as a patient, as a family member, you might wonder 10 years down the line, do I still have the transgene in my muscle? Or does my child still have the transgene in there, or is it diluted out in the way that I just explained to you? So while you are participating in long-term follow-up studies and registries, although there isn't really frequent follow-up, it's still important to tell us about health changes to be reported in the study so we can see any trends, so any hospitalizations, any serious illnesses. And sometimes I've found that I've had to really dig hard to find out this information. Sometimes I have patients, and it could be cultural sometimes, where they think that if they tell us that, my child had a pneumonia, that's going to hurt the drug, and we're not. We just need to know the truth so we can understand, and it doesn't necessarily mean that's bad for the drug. And if it is the drug that caused something, we need to know about that. So we really try to get accurate reporting to understand what the long-term adverse events might be. This will allow investigators and the sponsor to collect long-term effects of gene therapy and help us understand gene therapy. Finally, if you move, it's also important for those types of arrangements to be made. Perhaps another long-term follow-up study is a more appropriate site, and so that moving to another long-term site would be appropriate in helping to work with physicians or other providers that are local to them. So whenever possible, it's important that your primary physician is informed about any updates that arise during these study visits, and you may also need to transition some of your care back to a local physician who specializes in your disease, a neurologist, oncologist, etc. And the final thing I want to talk about is death. And this is an important topic to talk about because it is, possible you have a serious disease, and if you're undergoing gene therapy, that's something that's possible. And what happens if and when I die? We are going to want to discuss donating your body for an autopsy or some type of analysis, and it would be extremely valuable to help us understand more about the disease and the treatment. And you can really help a lot more other people with this disease. And this is something that we want to talk about early because it's not, sometimes it happens, unexpectedly. Sometimes it happens completely unrelated to the disease or the treatment. And then it's important to be able to understand what everyone's position on this. If you are the patient, we want to understand what your position on this is. And it's an uncomfortable topic. It's an uncomfortable topic for the doctors. It's an uncomfortable topic for the family. You don't want to discuss that because you want to remain positive and hopeful. But if it does happen, we don't want it to be a sudden rushed discussion where nobody knows what are your wishes. And it would be really helpful whether or not your death is related to the gene therapy treatment, for us to understand that beforehand. And so if you are the patient, sometimes it's easier if you bring up this topic. Certainly, I think everybody has a different comfort level with discussing this, but it's important. So finally, I just want to emphasize that you're important and your data are very important. If you participate in a clinical trial, it is the most rigorous scientific experiment, if I can use that term, okay? So we can understand what your body has gone through in the clinical trial. You would be one of the earliest patients to have received gene therapy just because you're in the clinical trial. And so you're a pioneer in your disease, which means that doctors and other patients are looking to you for guidance. And your data in these studies is extremely valuable to physicians, to scientists, and patients in the field. So please discuss with your doctor, what to do in the event of your death. If you're willing to donate part of your body, perhaps it doesn't even have to be everything. It could be specific organs. What would be helpful in advancing the field? And I thank you for your attention.
You can go ahead and stand up. Yep. All right, we'll invite the panel to come back up here, and as they do, we have one online question. If you are online, please submit your questions in the chat box. And then some reminders for those in the audience. We have a microphone in the middle of the room, so that we just ask to not touch the mic if at all possible to prevent spread. So I guess maybe we'll start. We have our panel up here. Thank you guys all for a wonderful presentation. Let's start with our first question from the audience.
Great discussion, by the way. With AAV and understanding that you only have one chance to be dosed, how do you feel about putting patients on studies that are first in human dose escalation, where you know the first dose is most likely to be the least efficacious? And how do your patients feel about that?
Dr. Goodspeed, they're all looking at all those. Dr. Goodspeed, yeah.
Is this mic working? No? Oh, here it is. There. Okay, if you don't push it, it works. Okay, I'll take this off real quick. So the way that we've thought about it is that we're not going in with the initial dose that's not effective. So at a bare minimum, that first dose is an effective dose. It just may not be the most effective dose, but it's the dose that is effective and thought to be safe. So I think that's how we have justified it in our center, and I'll pass it to others if they have other comments.
So I think we've learned to some extent from the spinal muscular atrophy clinical trial, the first study which really looked at systemic AAV gene therapy. And the first 3 patients were dosed at a lower dose, and I think that we've now started to understand perhaps what are the doses that are kind of the threshold for achieving the amount of expression that we need to achieve therapeutic levels. And I think that, moving forward, I've noticed that a lot of the studies definitely start at a higher dose than that study started, and really it's a dose escalation from a point which most people would agree should be an efficacious dose. Do you want to make any additional comments?
Sorry, so I've had the opportunity to be sponsor investigator on 2 trials now, and for my first trial, we started with what we thought was the minimal efficacious dose, and for my second trial, we were able to pick what we thought was going to be the most efficacious dose for all, and we're just planning on 1 dose at this point. But they're 2 very different diseases, and I think in the community we are learning more and more, and we're certainly feeling comfortable with higher doses, but I think it also depends on how broad or narrow your patient population is that you are studying. And I think we're learning now that doses for certain ages or weights or sizes may need to be variable, depending on whether you're targeting muscle or if you're targeting neurons. And so we're definitely very aware. We are certainly not giving anyone a dose that we think would not provide any efficacy, but we strive for safety first. And then, but we are willing to, we're learning as we go and always putting safety first, but really trying to minimize risk best we can and adjusting our strategies as we go. Hopefully that was helpful.
I'll just add as a former PI and now on the other side in industry is, you'll see in the rare disease world, though, you're also seeing smaller cohorts in the dose escalation. So we are trying to minimize the risk, but it does slow it down too, right? So we are starting with a minimally efficacious dose. Again, translation from non-human primates and other large animals, hopefully getting a better sense of things, learning from each program, but it does depend on the targeted organ and the disease state, the age range, and all of that. So I think we're learning from each other, and it's really important to share data across to understand it better. But we have to start with an MED and then go from there. But we are trying to minimize the exposure in each escalation. Because if we did go out, if no prior information going out with a higher dose and there's a significant SAE, there's no mitigation strategy. There's no way to move forward with that program either. So you're going to see a stepwise approach generally. But as we learn, you might see a more direct to one dose as well.
Thank you. I do have an online question that I'll read. One of the pillars of the Belmont report is justice. What is being done to ensure that financial and other social barriers do not unnecessarily exclude underserved populations, other than vulnerable ones, from clinical trials that could benefit them?
I'll take
Dr.
that
Lau, do you
one.
want to take that one?
That speaks to me.
Yeah.
So I think it's important to note that, as you're looking at a clinical trial, generally we do reimburse for travel. We reimburse for the relocation. As a PI, I was involved in relocating patients from different communities. And it's important that we reach out to all patients, regardless of where they are. And it's important that our communication with the patient advocacy community looks for diversity and is not to help those with barriers, with socioeconomic barriers. So, it's hard if you have a financial situation to work with your PI, work with your referring provider, but we reimburse. We can't pay for a patient to be in a trial necessarily because that's an incentive, but we do reimburse for travel, we reimburse for relocation. It's important that we put that into our patient engagement strategy or recruitment strategy. It's important to get a sense of what, to make sure that we are ensuring a diverse enrollment.
Thank you.
Sorry about that. This is becoming one of my passions as well. I think we always want to enroll as quickly as we can, right? And we know that there are some people out there who are more connected and just more savvy than others. And so I've been striving to make my coordinators more aware and my team more aware that just because somebody can't respond to us via email in the week time frame that we give doesn't mean that they're immediately off the table. Some families need more support, need more assistance, need us to reach out to them more. And it definitely takes a lot more team work and effort on our end, but I think it's very valuable to be able to get a more diversified study population. And so we're working on it, but I know we have a long ways to go.
Thank you. Would you like to ask the next question? Yep.
Hello, my name is Michael. I'm from Seattle. And I am a patient advocate, and one of the words that I heard today was that word patient. Sometimes I heard subject, but I didn't really hear it nearly as much, the word participant or even co-creator. And so my question really goes to in what ways for the clinical trial design process have you engaged stakeholders to help you co-create it so the end outcome is getting to things like travel reimbursement or a unique way of perhaps enrolling the study? Thank you.
So I'll take that first. So being part of industry right now, reason why I've joined is to have a bigger impact on the early process and research, right? And so what's really wonderful is that I involved patient advocacy in my practice from the beginning, being on research foundations and working with our patients and caregivers. And there are industry partners out there that start very early. It's hard because when you're preclinical, you're not guaranteed to go forward to the human subjects, to our participants. So we don't want to raise expectations, but on the other hand, we do need to rely on talking with patients and advocacy groups early to understand the disease. I work in rare disease. We don't know everything about that particular disease. We need to know directly from those who are affected by it. So while you're doing the preclinical work, it's integral, especially at where I work now, is we engage very early to have those conversations, to say, what is important to you or your child? How is the disease affecting you? Not just biochemically, not genetically, but how does it impact your daily life? How does it impact your ability to go to school, to work, or even just simply live? So I think it's important. I can't speak for all companies out there, but it's important that companies engage very early with the future participants as equals at the table. So I'm proud to work at a place like that.
I just thought I would make a little comment about the terminology. As a researcher, we're oftentimes highly discouraged from using the word patient, and it's not meant to be depersonify the patients. In many ways, it's an important reminder that it is an experiment, and as sterile as that sounds, sometimes it's important to realize that clinical trials, as much as it brings hope for treatment, there's enough equipoise, meaning that we have enough, unknowns involved in that it needs to be reemphasized that we are dealing in an experiment. And so then patients become participants or become research subjects. And so it may sound very, it may sound just kind of dehumanizing to use that term. That's not the intent, I think.
Next question.
Hi, I'm Susan Rudiger, and I'm the co-founder of the CMT Research Foundation. And you guys are all neurologists, so thank you for your work that you're doing, because I'm feeling very encouraged about the state of CMT coming, and you guys are paving some good roads for us. One of the things I also want to thank you for is your involvement with the patient advocacy groups. I saw you all had those kinds of disclosures. And my question to you is, as a research foundation, we fund some research, but not all. And I really want to hear from you, from academic and industry and clinic, what role do you want the patient advocacy organizations to play from a promotion or a neutrality? I feel like it's our responsibility to responsibly educate the patients, and we like to be very scientifically informed, but I'm nervous as trials are beginning to be thought about for CMT and gene therapies. Where do we draw the line, and what would you like to see us? How neutral or pro or con, or how would you like to see us communicate in a way that protects the patients, represents our scientific opinion, but yet is informative? This is a bit of a conundrum, and I'd love to hear your thoughts on that.
I love the question. I think it's a great talking point that we could probably spend a whole day discussing. One of the things that I'll kind of address a couple parts of that. A model that we've taken in Dallas is, much of that early preclinical work is funded by the foundations. They get that proof of concept data done, and as soon as there's a bit of a signal, we're trying to partner on the clinical side too, to make sure that we're getting both of those pieces tackled, or at least organized, and figure out who is seeing these patients. Can we increase access? Can we expand on anything that's already being done? But on the other side of that, what I really love that I've gotten to work with a few groups on is family meetings. So a lot of groups will have a scientific day where it's, the high level doctors talking to doctors, scientists talking to scientists, but also have a family day where you force us to speak family language and actually tell people and explain what we're doing, explain what those findings are, and empower people to come and hear it for themselves, but also in a forum where they feel empowered to ask questions and push and say, hey, but what about this other thing that you guys aren't studying? And that can also drive new research questions. It could drive us to think of new outcome measures to include. And so I think that dialogue is super important, and it has to happen. On the other side of that, I've seen some communities too starting to have more general symposia or talks at their symposia, kind of like what we're doing here. What is a clinical trial? What do you mean when you say that? What are these different phases? what is an IND and why are we speaking with acronyms? And so having some of those level setting opportunities too, and a lot of these are put up on YouTube now. So Simon's Foundation is one where I've given a similar talk to what I gave today that can be completely accessible, and you could even just point your community to have some of these educational pieces. ASGCT's Patient Outreach Committee is also working on a lot of material that is general material that's just good to help level set and get your community prepped for all of this kind of deluge of information that's about to come their way.
Just want to add, though, as a, working with patient advocacy, I think it's important to represent all the stakeholders out there and try to be neutral to a sense, right? To invite a variety of academics or, to have an open meeting of all the potential trials out there or the potential studies. As a physician in my clinic, I would always just represent all the trials out there, not just the ones I was running. And so I would hope that patient advocacy groups would do the same and just say, here's the information. If there's a clear benefit to a trial that's coming out, obviously you're going to promote that. But I would suggest try to remain as neutral as possible and work with everyone out there until there's a clear, a therapy that's working.
Next question.
Hi, my name is Leslie, and I'm a patient education liaison. And one of the questions I have for you guys is, what are you guys doing in your practice and your research to kind of help navigate some of the cultural barriers or, as we try to broaden our scope of getting more people into clinical trials, maybe some of the misnomers of information out there, and have you guys seen a setback with the challenges of having the pandemic and a lot of misinformation out there? I feel like from our end, we're getting so many different questions that we didn't get before, or so much misinformation that we hadn't seen in the past. And I was wondering if you guys were experiencing that and how you were navigating that with patients.
Yes. So to the, both for the previous question and the current question, I think I found that patient advocacy foundations have been excellent in terms of, for rare genetic diseases, I'll say specifically the FSHD Society has a very nice piece about COVID vaccines, and potential eligibility for gene therapy clinical trials. And I think helping to work both at an institutional level as well as on a patient-by-patient level, navigating that kind of conversation is critical. Certainly, it was evolving earlier on in the pandemic in terms of concerns regarding receiving the COVID vaccine and potential ineligibility for gene therapy, which currently, my understanding is that's not of a concern for currently commercially available COVID vaccines. But regardless, I think it's an evolving conversation and one that's important to have. I think also for this diversity, equity, and inclusion piece that's important to discuss is how do we as a healthcare profession increase diversity amongst us, both in terms of race, gender, and background, to help represent our patients, and whether it's speaking languages of our patients that are non-English speakers, as well as including interpreters, in-person interpreters, and potentially multiple interpreters if needed, to make sure that effective communication is reached is critical for these trials.
Thank you. And we'll take our last, go ahead. Can I have one thing? Sorry. I will just add a teeny tiny comment, but the only other thing that I will find is, so I work in the field of autism. There's a ton of controversy and snake oil salesmen in autism. And so what I will often do is I'll try to address any potential elephant in the room outright without them having to ask me about it, and say, some families ask me X, Y, or Z. Here's my thoughts on it. I don't know if that's ever come up for you guys. And just try to be really neutral while also tackling it head on and letting them know where I stand. I could see that being a beneficial thing, especially building trust or, they're afraid to ask a question, but if we know it's a question that tends to come up, I think it's good for us to ask the question and try to answer it, and then maybe that opens the dialogue a little bit too.
Thank you. Our last question? Yep.
I can wrap us up. Jody Wolf from Stride Bio. My question is about the long-term follow-up in gene therapy studies. So 5 to 15 years sure is a long time to be tracking trial participants. I was wondering if you could comment on some of the challenges that you've had, maybe what circumstances patients have been lost to follow-up, and maybe some proactive strategies on how to try and avoid that or to handle that.
So I guess I need to say something about that. So knock on wood, I've actually not lost anyone to follow-up, in the study so far. Certainly we have been creative in trying to make sure that that's doable. I have a reasonable number of spinal muscular atrophy patients who have actually come in from outside the country, Europe, even Russia, and our follow-up has been creative. We've used Zoom. We've had them, obtain laboratory studies locally. On Zoom, we've have our physical therapist, and we ask if they have a local physical therapist that, is able to do the maneuvers that we want them to do over Zoom, so that our physical therapist can observe and rate that. And so we've made it, about as accessible as possible, just for them to be able to do long-term follow-up for us, And so that's what those are the measures we've taken to try to make sure we don't lose anybody.
So it's not gene therapy, but we do have a paradigm for follow-up. So I was involved as a PI and physician, registries for enzyme replacement therapy in lysosomal world. So it was interesting because registries, there's limitations in the data that's collected, but I incorporated it at my visit. And so I'd made sure I had the new consent form, and I would gather the biomarkers and make it as easy as possible for their annual visit. These patients were following with me, and so I was able to collect lots of data. I had over a couple hundred patients in a variety of lysosomal disorders contributing to these registries that were put forth by the company. And then another form was a prospective data monitoring plan through Ultragenics that was as a PI. And that was a little bit more labor intensive, right? Because we were doing prospective, but we were offering clinical trial grade standard of care assessments that were reimbursed, but then we would share it back with the physician. I was the physician, so that was enabled our patients. They have to do these visits anyway for their standard of care, cardiac, pulmonary, brain, C-spine, all of that, and we would collect it under the prospective data monitoring plan. And it's really critical because we have to understand the long-term effects of any therapy that's a lifelong therapy, and that was one way to do it, and to reduce burden, so reimburse for the travel. And a lot of our patients with rare diseases are committed to understanding the long-term effects for themselves and for their children, or and to give back. So I think it's possible. We should be creative and try to figure out how to reduce burden. COVID taught us that we could do the telemed visits, we could do home-based assessments. So I think it's doable.
Thank you. And before we close this session, I thought I'd give our panelists one closing comment. So Dr. Goodspeed, do you want to start?
Sure. I just want to thank everybody for being here and going on this journey with us. I think what we've tried to impart is that we've learned a ton, but there's still a lot to learn and to figure out. And I'm just excited to see where this field is in 10 years. And we look back and say, gosh, I can't believe we were doing that 10 years ago.
Yeah, I think for me, the emerging emphasis on diversity, equity, inclusion coming into gene therapy clinical trials is nice that it's started to be touched on. I'm glad to see that it's starting to be incorporated into the week to come. So thank you.
I agree with all of that, and I think for me it's continuing just to look to the patient, the participant, understand not only the effects of the treatment, but the effects on the entire person, the psychosocial impact of being a participant, and understanding what that post-treatment phenotype, what is their disease state after? Is it, that cure, is such a difficult term to use here. So how are they responding after? And I think that's an important thing to think about.
Yeah, I think my comment, to an audience mostly of patients and family members is that, we recognize that this is a partnership that we have with you guys, and we're working together. We're going to do everything we can, and, as long as we work together, I think we can be we'll have a successful outcome for this.
I think I would just say I'm very grateful that I was able to be a part of this and that there are things like this happening now. Too long medicine in general has, been paternalistic, and now we're evolving, and I think that's going to really move things forward. The more that patients, families are involved, we're just going to be able to get the best care we can.
Well, thank you all very much. Thank you for those that are attending in person and online. I did see a question that came up for Dr. Shieh, so the person online who asked that, I'll pass it on to him. But thank you all. Thank you to our panelists, and we'll reconvene at 3:30 for the next half of our workshop today. Thank you.
That's probably not relevant for everybody, but I wanted to acknowledge the question.
Okay.
Yeah. So I passed it along.
Do I have to answer it right now?
No.
Okay. I don't even have an answer yet.
But I addressed it and I showed it to you,
Okay.
so
Okay.
I figured, it wasn't appropriate to ask. So.
There's a way for Allie to probably get that question to you, or in a way that we can answer it later.
Yeah, so let's just,
It's
that's a good
It's
idea.
not an easy question.
No. Yes, agreed. Well, great job.
Okay, thank you.
I loved your explanation of Lenti versus AAV. I thought that was a really great.
Yes.
And also long-term durability. I was like, oh, that's awesome. I haven't heard it explained that well.
Okay, thank you.
Appreciate that.
Yeah.
I was about to say, what about motor neurons? And of course you got it, the neurons. Yeah. Yeah.
I was looking all over the internet for the perfect figure to try to explain that too. So.
Yeah, it was really good.
Yeah, it's not easy to find a figure like that. So.
Yeah.
It's so simple yet.
Yeah. It just didn't exist. Simple enough, exactly. I thought it was, it's probably something ASGCT should look at for the patient communities, because they've got the differences between integrating versus not integrating.
A webinar on that.
Yeah. Cool.
Thank you.
You should give it. Yeah, that's what I was going to say. Can I volunteer you? Yeah.
Right.
Awesome. Are you sticking around, or are you heading out? Are you going to be around this week, or?
I'm leaving tomorrow night.
Okay.
Share with them abstracts.
Oh, cool. I don't run into it. It's fabulous to see you. So I do want to see pictures of your kids too.
There are a lot of them.
You've got, You're getting close. Yeah. There you go.
How many children do you have?
Oh wow.
My wife has 4. Yes. I'm around. I'll have a little bit.
Fair. You're busy. She's busy.
Yeah.
Oh, this is for
We should grab a pen.
I know. Next time I will get a pen, darn it. Oh, where are my slides? I must have. Kim, I'm going to slide you over a bit and put the computer back up here. There we go. I think that went really well. Oh my gosh, I was so nervous, and then I'm like, trying to they don't need to hear the intro, but then I'm like, probably rushed it a little too much, but anyway.
Okay, I'm gonna go get my pen too. Actually, I've got my slides. I always do this, I print all my stuff and then I don't use it, but.
like that work for Bill?
Yeah, here you go. Okay, yeah, I'll just email it to him right now. I'll just go sit here quick. Yeah. Actually, I wonder if I can see it on my phone.
I wondered too.
Actually, I might be able to. I can maybe just copy and paste.
Yeah. And then I figured.
Oh, here it is.
Yeah, perfect. And I figured there's probably a way to send it back to that attendee.
Oh, I wonder if we could just get that. He could go on the discussion now during the session and respond.
Yeah. He said it's going to take him a little bit to, just because it's such a specific.
Do we ask the person to email it to the info box? I don't know how to get their contact info.
Yeah.
Well, I'll write down the question and the name and figure out how I can get the contact info. Because I can probably look it up in the registration info. Oh, yes. Is all of this alright? It looks a little different on here. Okay, I got it, and I'll figure out a way to get it to him. Yeah. And I think you guys probably know it was pretty much on the same. Just Julia's presentation will show up on there, and then she's on, so she will show up on the screen to chime in for the panel. Okay. So.
Do you want me to acknowledge that this is a pre-recorded and then she'll be able to just...
You could. Yeah, I mean, they'll know. I mean, when it's...
Yeah, when she has a different... That's true, when she has a
But if you want to do a normal, like, introduction and then they'll play it, that's fine too. Like, introduce her somewhat.
I found it. Yes. Yeah.
Perfect.
How do I say your last name?
Sade?
Sade.
Yeah, that works.
And is it an S or SH sound?
No, it's an S.
It's an S. Sade.
That works. It's actually Sade, but that's like an
Oh, I can say Sade.
Oh, I like that. That's pretty.
Sade. Finnish also pronounce every vowel, even if they're double vowels. So
Yeah, it's make up for a letter that
Okay, for a long
doesn't
period
exist in
too.
English. Oh, good. So I could start too late. Okay, started right on time, and I thought, oh, no, wait, it's catching up to the online. Good.
No, I think you're okay. Good. They're just starting, like, at, the time of starting the session. They start
Okay, they just
session.
start. Okay,
I think.
yeah.
So I think that's just how it'll run now. It's just the session starts, so once the person is talking, they are seeing it. I thought you were great. I thought that went really well.
I thought it went really
Super
well.
interesting.
Really great. Good.
Yeah.
I think so.
Cool. All right, well, I'm going to get out of your hair and... Hello. Hi, thanks for coming.
Nice to see
Oh, Emily, good to see you. I don't, with the mask on.
Oh, yeah, here, I'll do this. That's a bunch of the, yeah.
Good.
I love it. I was admiring your dress from afar.
Very cool. I was too.
Oh, well, thank you. And I didn't upload slides because I'm inept. I was in Europe and I couldn't figure it out. And then we migrated to our service to the cloud, and then my computer was like, I'm not doing anything more. So I'll just talk from
the heart. You'll be fine.
I'm Dima, by the way. I'm a child neuromuscular neurologist. I work with Diana. Nice to meet you.
You work with Diana?
Bharucha, Carson, Donovan.
Oh, and Carson.
And now I'm back working with Kathy Matthews.
Very good.
Kathy was one of my mentors when I did the clinical trial methodology course. She's so lovely. She is.
Yeah, Kathy's wonderful. I serve on a couple of BSMDs with her. She's everywhere, right?
Yeah. He's a great mentor.
Yeah. All right.
All right.
Do you want us all up here?
Yeah, what did you do? I know we broke it up because it was so tight before, but since there's three of us, should we all stay, or do you want us to go back and forth?
Yeah, maybe just stay up here.
Yeah. Okay. I love your shoes too.
You're adorable.
Yeah.
And I'm Kim. I don't think I officially
No,
said that.
Kim, nice to meet you. Really nice to meet you. All right.
All right, I guess we can. Do you want to sit here, or do you look good right there? Okay.
I'm good here, and then I'll talk about them.
Perfect. Let's see here.
And Allie took that question down and
Perfect.
try to figure out what to do about it.
Okay, good.
She's like, I don't know exactly what to do, but figure out something.
Good.
are you? I didn't do slides. Oh, that's fine. No, and it's totally my fault. I was in Europe and I tried to upload slides. In the middle of there, they migrated our servers to the cloud, and my computer was like, I don't know.
You know, I was going to blow over this slide.
I have a few notes, and I'll just tell the story, Allison, if that's
But since that question came up from the gentleman, I mean, I don't want to brag, but I hate to be, you know.
Yeah.
Anyway, I might just bring it. It'd be really cute.
Okay, well, no, it's fine. Okay.
No worries, I just didn't, I wanted to make sure that there wasn't some like.
Yeah, no, my mix-up completely.
It's no worries.
Okay, great, thank you.
Are you here for the week? Yeah, this one?
I'm going home tomorrow night.
Okay, me too.
And then do the rest virtually.
Yeah, that's plan as well. I thought about staying longer, and then I thought about it, and then I thought, let me check hotel rooms. I checked hotel rooms, and it was like, oh, no, nothing. So I'll just go home tomorrow.
We end up streaming most of it anyways because of all the parallel stations.
Yeah, that's true.
It's a good conference, though.
Yeah. This is my first time, actually.
Right. Me too. Is it yours too? Yeah.
I may have watched it last year. I can't remember.
Yeah.
This is my first time. I get it mixed up with World.
Yeah.
Similar content for me.
Yeah. I missed World this year too, so. Yeah. In and out. It is nice to be back in person. It's just a big shift. Nice to see everybody.
In the meetings I've been in, no one's been wearing masks.
What's that?
No one's been wearing masks. And I just traveled, I was in Amsterdam for about 7 days until last night. No one wears masks in Amsterdam, and on the planes they're not wearing masks. So it's really kind of
They
interesting.
ditched the masks long ago.
Yeah,
Or I
yeah.
don't know that they imposed them.
Yeah.
It just feels weird.
I know we got so used to wearing them that it was like none. Yeah. I kept catching myself. Anyway, grab my walker before we started. That's a good idea. Anybody need anything else while I'm down?
No, thank you.
To doing the pre-recorded, or even if you can't really see anybody, it is weird. I was totally shaking. I'm sure I'll be shaking again once I see it here again. The reason I wear tall necks because I turn red. I blush really bad. So if I'm ever speaking in front of people, I wear a high neck or a neck. And things that don't just sweat because I also sweat.
Yeah.
Hopefully these aren't hot nights. They probably are.
Doesn't help when you have a heavy or jacket on. And,
Hit or miss at those angles. You either freeze or it's a little hot under the lights up here. right, if everybody wants to trickle back in and take your seats, we'll try to get our second session started off. I'll go through a couple of little announcements and reminders. So for session 2 this afternoon, we are going to be covering the risks, challenges, and hopes associated with clinical trials. And so this part of the session is really going to bring in more of that human side and patient side, and we'll get to hear from a couple of people on the front lines, including physicians as well as families. And so Emily McGinnis is going to start us off with a bit of a caregiver insight study that was done. Then Dr. Sade is going to go through how she thinks about clinical trials and weighing those risks and benefits with participants. And then we get to hear from a couple of families. So Pat Furlong from Duchenne Muscular Dystrophy and Julia Vitarello from CLN7. And her talk will be a pre-recorded talk, and then she's going to get to join us for the panel discussion at the end. And similar to session 1, we're going to hold all questions till the end and get to discuss everything together as a panel. And what we would love for everybody to do is, if you're in the room here, go ahead and make a line at the mic when we get to that panel session, and just try to refrain as best you can from touching the mic and keep your mask on while you're asking your question. If you guys are tuning in from home and virtually, there's a chat box that we'd love for you guys to put any comments or questions into that chat box, and we'll go through those there at the end. Okay. And then one other announcement for a little bit of housekeeping. To follow this session, we will end promptly at 5 p.m. Eastern Time. And we have a reception that's going to follow the conclusion of this symposium. So from 5 to 6:30 p.m. on the 3rd floor here in the Convention Center. All speakers and attendees are welcome to attend, and we're really looking forward to seeing you guys there and continuing the discussion or talking about something completely different. So we will get started with Ms. Emily McGinnis. So Emily is a Chief Patient Officer and Head of Government Affairs at Taysha Gene Therapies currently, but she's got a long-standing history of patient advocacy, especially in rare disease. She led patient advocacy and government affairs at Avexis and was instrumental in the launch of Zolgensma, where she played a key role in collaborating with advocacy on newborn screening development, as well as implementation efforts to rapidly diagnose spinal muscular atrophy patients in need of treatment. Today, I'm honored to partner with her as a co-chair for this workshop, and she's going to kick off our session 2 of Risks, Challenges, and Hopes with her talk, Caregiver Insights on Clinical Trial Participation.
Thank you. I feel like the gentleman who asked the question in the first session from Seattle set me up really nicely for my talk today. I have the honor and privilege to work with a number of not only advocacy groups, but also patients and caregiver in the rare disease space. And I'm not going to tell you a lot about Taysha, but we call our caregivers, patients, and advocacy groups ally. And that's really what Taysha means. It really represents our commitment to the people that we work for and the patients that we work with. So really our aim is to be an ally to the rare disease community, because together we can accomplish what has never been done before. So today I'm going to highlight some research that we have done at Taysha in the preclinical stage in preparation for gene therapy clinical trials. But first, I want to state that this presentation is dedicated to the caregivers who shared their experiences, hopes, and fears with us, and to their loved ones who are unable to speak on their own behalf. We hear you. So just to set things up, we at Taysha have really embraced the idea of patient-focused drug development. And that started with the FDA back in 2012. And the initiative was to bring together industry, regulatory authorities, to really bring the patient perspective into drug development. And we believe that it's critical, as does FDA, to not only provide education for regulatory authorities, educating us as drug developers, really with context and background about the disease, because it's the patients the families, the caregivers who are the experts in the disease. And oftentimes, as you learned earlier, there's really not a lot of natural history study data to inform clinical trial development. So discussing and learning from patients and advocacy groups can inform clinical trial design, including the selection of patient-reported outcomes, what are the outcome assessments, and what are the primary endpoints that we'll use in a clinical trial. This also can help support regulatory decision-making, especially the risk-benefit assessment for a new treatment or a new drug candidate, or gene therapy candidate in our case. So because in our case we deal with mostly pediatric patients, and it's the caregivers that are experts living with the disease, we capture these experiences again in the preclinical setting, really trying to understand the disease. And what I'm going to share with you was submitted and shown at NORD last October. So here's how we worked, and this is again covers a number of different studies that we had done. So we engaged with rare disease caregivers, specifically in different therapeutic areas, a total of 60 at this time when we cut the data. So we looked at SIRF1-associated Leigh syndrome, CLN1 Batten disease, GM2 gangliosidosis or Tay-Sachs and Sandoff disease, SLC13A5 deficiency, and also Rett syndrome. We collected insights through in-depth interviews, online surveys, and then a 2-week online discussion forum, which was kind of Facebook-esque, where caregivers could interact with each other, but also individually, again, very protected from a privacy perspective. And then we finalized the group with a 2-hour focus group so that we could take in all the learnings that we learned before, but then also ask more in-depth questions with folks via Zoom, which was really great. So what we learned, as you can imagine, each therapeutic area is unique. But we did find commonalities in these 5 disease states that I'll touch on today. And as I mentioned, we asked a lot of things, including what's the journey to diagnosis, but really our goal was to think about 2 questions. What are the most impactful symptoms to your loved ones, and then what would be a meaningful improvement? So first and foremost, communication across all of these diseases was important. It was challenging to understand their loved ones. It was also a source in distress for their family member. And second to that was motor issues. Motor issues often restrict their family member or their child's independence with school or other things in the real world, including contributing to the caregiver burden, always needing someone to be there for their child. And then we also asked, well, if you were to participate in a gene therapy clinical trial, what would be a meaningful improvement? And again, in communication, just the ability to potentially improve communication, using assistive devices, verbal yeses and nos. And then for motor issues, things, could therapy potentially impact the daily life, the ability to walk independently or sit or stand or play with their peers. So we also wanted to understand, what are the desired outcomes? And we found that for the most part, caregivers welcome the possibility of slowing or delaying disease progression. They also, talked a lot, as I mentioned earlier, about symptom relief or improved quality of life rather than prolonged life. From a risk-benefit perspective, the caregivers talked a lot about, where their child was in the stage of disease progression, and realized that the outcome or the risk-benefit was related to where they were at with their disease or their disease phenotype. And then the other consideration was what would as we learned earlier, that this is, in being in a clinical trial, can have an impact on their quality of life and eligibility for other trials, which I'll touch on in a bit. So we learned a lot, and I know I'm summarizing this really quickly, but and I actually am thrilled to be part of the patient outreach committee with ASGCT because I feel like we're tackling a lot of these topics, which is great coming from a third party. The caregivers wanted a lot of education, and this is, of course, are things that we fed back to our advocacy groups because patients and caregivers wanted to understand the mechanism of disease and what are the potential targets for gene therapy. What is the scientific basis of gene therapy? What would be the commitment of participating in a clinical trial and the potential risks and benefits? And then there really was a lot of desire to understand what is the clinical development process and the regulatory process, which hopefully we've touched on a lot of that today. And then from a support perspective, families also would like support with travel and planning and expense reimbursement, and of course accommodations, including accessible considerations. So I'll just wrap up here. Again, it was surprising to us looking at 5 really unique diseases, but that there are commonalities that we can think about as we're planning future clinical trials. Again, meaningful improvements focused on reducing discomfort, increasing independence, and improving the ability to interact with their families and peers. And these findings did suggest that communication and motor function should be explored when developing clinical programs for rare genetic diseases. Again, we take these learnings not only to inspire our colleagues within Taysha, but also we'll share them, with our regulatory team, our clinical team, anyone working on the clinical study, so these insights can help with developing the clinical trial protocol. But these learnings can also help us with clinical trial recruitment, training of the clinical trial staff, and then really what services do the families need when they do participate in a clinical trial. So that's all for me. Thank you.
Thanks so much, Emily. That was a great kickoff to this session. So up next, we're going to have Dr. Dima Saadeh. So she is a child neuromuscular neurologist and a clinical assistant professor at the University of Iowa Hospitals and Clinics. She's an investigator on several pediatric neuromuscular clinical trials, including gene therapy trials for Duchenne muscular dystrophy. Prior to joining the University of Iowa, Dr. Saadeh was a clinical research fellow and a staff clinician for the gene therapy at the NIH in the neuromuscular and neurogenetics disorders of childhood section, where she was an associate investigator for the giant axonal neuropathy intrathecal gene therapy trial and the systemic gene therapy trial for X-linked myotubular myopathy. Thank you. I had a blank on the acronym. Apologies. We're delighted to have her here today to talk with us about weighing the risks and benefits of participating in a gene therapy clinical trial.
Thank you. Thank you for the opportunity to present. So I'm just going to go over some of the discussion points that I make sure to go through with families when talking about participation in gene therapy. And just to frame this talk, this is mostly about AAV-mediated gene transfer. And I find this graphical abstract illustrates it very nicely. There is currently AAV-mediated gene transfer in trial for many neuromuscular and neurogenetic conditions, as illustrated here. So the promise of gene therapy, it is really exciting because in theory and in an ideal world, this could be a one-time treatment that delivers a normal or intact copy of a gene that could then address the root cause of a disease and start expressing a normal protein, which gets us a little bit closer to a cure, which is the word that we don't like to use. And this has led to approval of gene replacement therapy intraretinally for retinal dystrophy in 2017. And then more recently, in 2019, the approval of gene therapy for spinal muscular atrophy type 1. Now, these just going over clinical trials in general, and I don't think I need to introduce the different phases of a clinical trial to this crowd, but I will focus on the fact that although we say gene therapy clinical trials, it's not exactly a therapy. and we hope for a cure, but it's not a cure. And there's a lot of unknown, and that is why the trials are designed and conducted. And so when somebody is enrolling in a trial, most of the time, we don't know what the optimal dose is yet, what all the possible side effects can be. When you're enrolling in a trial, it could be open label and you know what you're getting, or you could be part of a randomized, blinded study where you may not know when you're receiving the gene therapy. And then trials can be unpredictable as they evolve. Some trials can be placed on hold, and then although you were already enrolled, may not get a chance to participate or be dosed. Some trials may enroll healthy siblings as control, and then increasingly trials are requiring full documentation of immunization, which can be a touchy subject. And then when talking about clinical trials, I think it's very important to understand what is the prospect of benefit, meaning what is the expected benefit from that trial. Do we hope for a slowing down or delaying of disease progression? Is there hope that it's going to halt disease progression, result in partial recovery or full recovery? What specific tissue is that trial, that gene therapy targeting? And what is the anticipated or thought of durability of the treatment? So it's very important to periodically ask investigators about what is the realistic extent of improvement based on knowing or understanding the disease, the preclinical data, and then the ongoing trial information. And then there's a few points that are very peculiar and specific to gene therapy. As of now, in most of the trials, it's a one-time only event. So if you are enrolled early on in a trial and receive a low dose, and then later on we learned that a higher dose is more efficacious, that's it, you cannot be redosed. But alternatively, you may be dosed at a higher dose, and then that is then found to be associated with increased risk, but you've already been dosed at a higher dose. It is, for now, irreversible. There is currently no way to turn off gene therapy once it's started or fine-tune it. And so one thing that is still unknown is, are there long-term risks associated with overexpression? And then what we're increasingly finding out is that preclinical trials in animal models are not always accurate models, especially when it comes to how the human immune system is going to react to gene therapy. So then moving on to safety in gene therapy, and there's a lot of unknowns, but I've divided it into a few categories that are emerging in the field. So the first safety concern is an immediate or acute or subacute immune response against the newly introduced AAV vector. And if you think of a viral infection, you get infected with a virus, it gradually replicates and increases its number, whereas in gene therapy, you're receiving a bolus, a very big, high concentration at once of viral particles. And your innate immune system, that's the body's first line of defense that reacts regardless of whether you have antibodies against the AAV, is expected to react to it, to fight it off. And so that can lead to adverse events such as fever, vomiting, cold-like symptoms. It could also result in severe inflammation and can be fatal. And then one emerging side effect in several different trials is thrombotic macroangiopathy, and this is a syndrome where there's destruction of red blood cells, decrease in the number of platelets that are important in clotting, in blood clotting, and then micro clots in several organs leading to organ function. And so to mitigate that, most, if not all, gene therapy trials have steroids that accompany the gene therapy. And that is meant to suppress the innate immune response to prevent that reaction or to dampen it. But with steroids, you also get an increased risk for infections. Some trials have built-in additional immune prophylaxis, and then in several trials now, there's closer monitoring and a low threshold to add additional immune suppressive agents. Another well-recognized gene therapy safety concern is liver toxicity, mostly from liver inflammation. Your liver is the filter organ. Even if the gene therapy is not targeting the liver is still going to see high doses of it, and it's going to react to it. And so what the main mitigation strategy is to dose with steroids for as long as needed, and monitor liver enzymes and make sure they don't rise or remain within a controllable limit. And then there's also the more recently recognized transgene-directed adaptive immune response. And so if you think of gene therapy as a mini transplant of a protein, if you have specific types of mutations where you don't make any protein, or you make a defective protein, then your body may not recognize the normal construct or the normal protein as self and may attack it. And sometimes you can predict whether this reaction is going to happen based on the mutation type. The possible consequences may be reduced effectiveness or reduced efficacy over time, lack of benefit, or even worse, damage to target tissues, and that has been seen with immune reactions against muscle and the heart. And so mitigation strategies can include prophylactic immune modulation for patients with specific mutation types to ensure immune tolerance, reactive immune suppression should such an event arise, but also exclusion of participants that have specific mutation types that are predicted to cause an adaptive immune response. And then one of the things that have emerged in the last few years is that sometimes this exposure to gene therapy may uncover a previously, that's the next slide, but then so with gene therapy, you may uncover individual susceptibilities to exaggerated immune responses. And these are very hard to predict. And if you think about it, it's kind of in the same category or kind of similar to why do some people have such an exacerbated, dramatic immune response to COVID or to the flu. And it's hard to predict. And through that, we're learning more about the immune system. But I know there's been discussion about doing whole exome or whole genome sequencing in all trial participants, either prospectively or retroactively. And then that's what I started talking about at the previous slide. The gene therapy can precipitate uncovering an aspect of a disease that remains elusive despite careful natural histories and several animal models. And that can have severe consequences. And I'm not going to go into the details, but if you've been at other presentations and talks and meetings, this is what is thought to be the reason behind the 4 deaths in the gene therapy trial for X-linked myotubular myopathy. One thing to keep in mind, too, is that with gene therapy, you alter the natural history of the disease, and so clinicians need to be aware of that and keep an open mind. And then there's other safety considerations, so all the complications from immune suppression. Integration and carcinogenesis does not seem to be a big threat in the neurogenetic space. And then there's the risk of dorsal root ganglia toxicity that's been observed in non-human primates, but also in one subject in an ALS, AAV-mediated gene silencing, but it hasn't been seen in other trials. And then there's more that may be uncovered over time. And then another thing to keep in mind is the burden of trial participation. So aside from meeting eligibility and receiving dosing, it comes with big number of procedures and schedule of events. And so most of those trials require admission and or temporary relocation at the time of enrollment. And then there's several follow-up visits in person, mostly in person with some adaptation with the pandemic, within a specific time window in the first year. And there's always the question of who can accompany the participants, and then families have to reorganize the whole family schedule and work schedule in order to be able to meet these. There's frequency safety monitoring in the first year. So participants undergo several repeated clinical assessments, blood draws from an indwelling line or from repeated phlebotomy. And that can also result, although we're very mindful of how much blood is drawn, but that can result in anemia. And then other procedures that may be trial dependent. And then also the efficacy assessments, and they could include activity monitoring, PT assessments, caregiver and clinical rating scales, and also in some trials, procedures such as muscle biopsies. This can have impact on siblings. If a sibling is similarly affected and one of them is enrolled in a trial, then parents may need to separate them to prevent seroconversion of one sibling that may then preclude them from participating in gene therapy. Gene therapy trials have a required long-term follow-up from the FDA, so up to 15, as long as 15 years, the first 5 years in person on a yearly basis, and the next following 10 years either in person or with questionnaire, so that we can learn about long-term adverse events. And it does take a toll on trial participants because it's a lot. And throughout all of that, there's a lot of uncertainty. And then one thing, too, that I talk about with my patients is that we clinicians in those trials try to engage the patient's local teams and local care providers. But sometimes you may find yourself in a situation where in an emergency room or with a new provider that may know very little about your child's condition or the experimental therapy that they've received. And this is where the care team, the research trial team can also weigh in. And then there's two scenarios that come to mind, and that's a discussion that I've had with some families, is when there are no other options, do you, besides supportive management, do you really have a choice? When we consent patients, we say, well, this participation is entirely voluntary, and you could choose not to take part of this. But then some parents express that they feel like they don't really have, in face of a disease that's relentlessly progressive, they don't really have many options. It's not a real choice because of the existing duress. And sometimes you think, well, should we wait a little bit longer and learn more about what the optimal dose is and what the safety profile is? But you may lose eligibility in the meantime. If it's a neurodegenerative disease, you may lose your window of therapeutic opportunity. And then in another scenario, you may have other options, some approved therapies versus several different trials to choose from. So how do you pick one? So it's important to know that some gene therapy trials require treatment-naive participants. Or, for example, for some of the Duchenne gene therapy trials, if a child is on FDA-approved exon skipping therapy, then they need to wash out from it before they can be considered as eligible for the gene therapy trial. But what if there's other trial options that are not gene therapy? They can't really compare side to side and maybe potentially safer. Which one to pick then? And then if there's several gene therapy trials for the same condition, again, like in Duchenne, which one to choose from if you're eligible for more than one. So going back to where we started, weighing the risk and burden against the benefit of participation. And really, there's no right answer. This is a very individual personal risk benefit assessment because trials are experimental, and although they bring along a lot of hope and promise, they also come at a high risk. And the trial participation is tedious and may come with no guarantee that the participants themselves are going to benefit, but they may be contributing to advancing the research and therapies for future other patients. And with that, I'd like to thank you.
That was amazing. Thank you so much, Dr. Sade. So up next, I have the pleasure of introducing Pat Furlong. So she's the founding president and CEO of Parent Project Muscular Dystrophy, PPMD, which is the largest nonprofit organization in the United States solely focused on Duchenne muscular dystrophy. She's a nurse by training, and when her two sons, Christopher and Patrick, were diagnosed with DMD in 1984, she dedicated her life's work to advancing research in DMD and ultimately to find a cure. Along with leading PPMD, Pat speaks about Duchenne and related topics at conferences. We're delighted to have her here today, and is an active board member with the National Health Council. She is also a committee member of the Clinical Trials Transformation Initiative and serves on the Data Safety Monitoring Boards for both the Rare Diseases Clinical Research Network and Cooperative International Neuromuscular Research Group. Today, she's going to address the participant challenges in gene and cell therapy clinical trials.
Thank you very much, and I'm really delighted to be here. And I have to say I have no slides because of my own inability to perform in that area. I've been in Europe and couldn't seem to get my act together to get slides uploaded, so I apologize. So I thought I'd tell you a story. First of all, there's a language of medicine, and there's a language of science, and there's a language of illness, and they're quite different. And patients, or subjects, or people, or children with a diagnosis, and their parents learn all of them. They're exposed, once a diagnosis occurs, to all sorts of inputs, as simple as Dr. Google, that says whatever Dr. Google says, to as they become really involved in a community, they hear other words. They hear words from industry. They see forward-looking statements that are very forward-looking statements. They listen to investor calls. And investor calls have also very different language than these patients and families know. They hear about gene therapy, one and done. They hear about gene therapy in Duchenne muscular dystrophy as mutation agnostic. So let me set the stage for Duchenne. Duchenne muscular dystrophy is diagnosed in childhood about the age of 4 to 6 years old. It's a progressive neuromuscular disease. These babies are born and they look normal. They have delayed milestones, but over the period of the next 20 years following diagnosis, they will lose every bit of function they have. First they start falling, then they lose ambulation. Then they can't lift their hand to their mouth, and then they need non-invasive ventilation, and the mean age of death is 30. So the good news about that is we've been able, through use of non-invasive ventilation, cardiac medicine, steroids, to be able to change the mean age of death to 30. The bad news about this is we've been able to change the mean age of death to 30 because at the age of 15, most of these boys cannot lift a hand to their mouth. So they have no privacy. There's a loss of self-esteem. We use chronic steroids, which cause all of the steroid effects. So it's a very difficult disease. And when we see and hear about gene therapy, we heard about Zolgensma, right? SMA1 babies who were anticipated to die at 48 months. The primary outcome measures were ventilation and death, which are easy things to diagnose. And in Duchenne, you have a series of what I consider little deaths: the loss of ambulation and its ripple effect on the family, the loss of the ability to feed yourself and its ripple effect, and it goes on and on. So, gene therapy studies are underway, several different companies, and we are incredibly enthusiastic because in Duchenne, we've got one of the largest genes in the human genome, 2.4 million base pairs, 79 exons, and about, I don't know, lots of mutations, maybe 1,000 of them across that gene. So what the concept of gene therapy in Duchenne is we're going to give you, through this gene therapy using an AAV and a promoter, we're going to put into this AAV serotype, not that full length, but a smaller version called a microdystrophin or a mini dystrophin, depending on who's talking about it. And so for the parent hearing that mini dystrophin comes back, it feels like the Holy Grail. And we just talked about, and you heard these talks about low dose, high dose, or lowest therapeutic dose. But to me, as a parent, low dose when you're going to a high dose says something to me about there's a better thing out there. And maybe my son isn't going to get it. Also, you heard Dr. Shieh talk about the turnover here with muscle. It doesn't get into the satellite cells, so there's the durability of effect is unknown. And in the investor calls, we hear 5 to 10 years and maybe a lifetime, but maybe not. So we at PPMD did some benefit risk studies, first of all, before the studies began to say, how do you feel about benefit risk? And the parents were so enthusiastic about it and said, we'll take on the risk. When we said the risk of death, we had this whole thing that we worked with a professional group to make sure our study was rigorous and helpful, and we found that there was a willingness to take on considerable risk. We also did caregiver studies and young adult studies, and they too, those considerations really were concordant. They agreed. They were willing to take on risk. And it seems, at least in our studies, across the trajectory of this illness, that the later you get, the more impaired you get, the more willingness you are to take risk, both by the caregiver and the person themselves. So you're entering a clinical trial. You've heard from Dr. Google. You've been on the investor calls. You've heard one and done, which is the ticket out. But this child hasn't had microdystrophin or anything else since birth. So if we're looking at a 4-year-old, he's got considerable damage. So what can we expect? Depends on the language. We had a mom that was going into a clinical trial in gene therapy, and on her way in, the language we use is important. She said to her son, this is magic medicine. It's not. And when he was taken away from his school for repeated tests and tests and tests, and he didn't play the Minecraft business, whatever they're playing, that I don't understand. He felt more and more disenfranchised from his group, and he wanted to be out of a gene therapy experiment. So there's all sorts of complications and risks and benefits to the thinking behind gene therapy. The informed consent, parents have said it's neither informed nor consent when you have no options. It's what you need to do for your son. The assent, how do you say to a 4-year-old, what you're getting into? They look to mom and say, okay, if you or dad think that's right, I'm going to do that then. We've done benefit risk studies over time. We believe in gene therapy, but it isn't without risk. We also have to learn a new language. You've just heard of some of those. The immune response to the transgene. I can't even imagine how some parents look at those. The promoter, the microdystrophin, even the mutation. And mutation for some people lands so hard it feels like they're some sort of alien being. And then when we work with our companies, they were saying to us that we're going to invite patients to the table. Well, we kind of said to them, it's our table. You're coming in, and let's do our best to partner together and make sure we do well for these children. So what we've done along the way is we're writing guidance for FDA so that we can really explore with our patient community what the benefits and risk and what inherent knowledge we have about this disease. How well is it characterized? What does care look like? What happens in the outcome measures? What are we looking about? And especially in a clinical trial with a 4-year-old, we can't do every outcome measure. This child's going to have a biopsy at baseline. Then he's going to go through all of the exams to understand if you're a good candidate. And then he's going to get his blood drawn to see if his antibodies to the virus, which may come 2 weeks after that screening. So it's like a game of chutes and ladders. Gene therapy, yes. You have antibodies? No. You are too old? No. And then what's next? And so finally, what we're thinking about now, and I think Dr. Shieh, if he's still in the audience, can talk about this, we have a growing group of about 25 parents whose children have been involved in gene therapies who are saying, what's next? If the durability is not going to last for a lifetime, what are we collectively as a community going to do and think about and add on therapies, and how do we do that? And how do we do trials and registries for children who've been in gene therapy studies, not just to look at how is that gene therapy doing, but how can we preserve and protect whatever was gained by this study so that these boys live longer, better lives, and we close that gap between 15 years old, loss of all function, and mean age of death. So we close that down. So we protect them so they grow old. So we've also begun a community advisory board in Europe. So because it's our table, our community advisory board invites industry as early as they want to come to us, and we have a group of 15 parents represented of 15 countries, Eastern and Western Europe, Israel, China, Australia, and we talk to them about clinical trials and burden, and especially what's next. Will we be okay? Will our son be okay? And how are we going to partner together to make sure whatever we receive from this benefit and this hope and this opportunity is real, is measured, and is helpful to go on to the next step. Thank you.
Thank you so much, Pat. That was such a powerful story. Okay, so this is our final talk before our panel discussion, so I want to just do a quick housekeeping reminder for anybody watching online to remember to put in your questions to the chat box so that we can have those and go through them later. So our final speaker is Miss Julia Vitarello. So she founded Mila's Miracle Foundation in 2016 upon learning that her 6-year-old daughter Mila had Batten disease, a fatal genetic condition with no cure. In a race to save Mila, Julia's collaboration with Dr. Timothy Yu from Boston Children's Hospital led to the first ever drug tailored to just one person, affectionately named Milacin. After showing great promise in the first year of treatment, Mila's disease slowly progressed. And in February 2021, Mila's spirit left her body. But driven by a sense of hope and duty, Julia is on a mission to open up the field of individualized medicines for many more. So we'll move to her talk, and then she'll join the panel at the end.
Thank you, everyone at ASGCT, for inviting me to speak today. I'm sorry I can't be there in person, but I don't want to share my COVID with any of you. Today, I want to tell you a little bit about my daughter, Mila.
Here we go. I'd like to start by thanking all of you for coming to help celebrate 2019's Rare Disease Day with us. I'm going to be telling you a little bit about a young girl named Mila. Mila was a typically developing child, precocious by many accounts, until around age 4, when she began suffering a mysterious decline.
She could have really been anyone's child. I think it's one of the reasons why Mila's story has gotten quite a lot of attention around the world. She went on hikes for 2-3 hours at a time. A group of people came up to her and they said, "Hey, little guy, aren't you cute?" And she said, "I'm not a guy and I'm not cute, I'm dangerous." You know, she was...
At that time, she was beginning to show some unusual symptoms, including pulling books up close to her face, having trouble discerning the letters in low light. At age 5, she began stumbling and unusual motor movements of her feet. Things became much more serious in a very short period of time.
The ophthalmologist, his advice to me was to chill out and just relax. That was 6 months before she lost her vision completely. And they did an incredible amount of tests that ended up in a diagnosis of Batten disease. No cure, no treatment, and it was 100% fatal.
We said, this is a family that's in need. We called up the family, and I introduced myself, and I said we'd like to help. We had been thinking about how to roll out whole genome sequencing for patients for quite some time.
I will never forget the phone call I got. Our geneticist said, "Tim has something he wants to tell you." He actually thought outside the box. The day that she received her first dose, I walked through the hallways of Boston Children's Hospital for a few hours. And I thought, oh my gosh.
Hi, Mila. Am I the big leader?
People told me that it was impossible. They told me that it would take 10 years and that it would never be in time for Mila.
This is sort of the best confluence of interests where, a family's desire to help their child also helps many other people in this very small rare disease community.
This was an entirely new drug targeted to one child. Mila's drug was called Milacin. It's named after her. This was an opportunity for me to tell Mila's story, to show the world that together we can race to create a new treatment path that could save the lives of children across hundreds or thousands of diseases. My mission, together with Dr. Yu, was to apply what we did for Mila to many other children.
We've seen a revolution in the way that we can make diagnoses. We can sequence genomes almost at the drop of a hat now. But to actually develop a therapy in this short period of time, that's a first. The question is, how do we scale this out to as many patients as possible in as safe a way as possible?
Why can't we have 1,000 drugs for 1,000 different kids? Mila is the first. She had to break that ice to say this is possible. Now we are entering another unknown territory, which is how do we rehabilitate a child like Mila that's lost so much?
It doesn't mean that everything is magically solved. It doesn't mean that every patient like Mila will have a customized drug for her tomorrow or the next day or next year. But it shows that when we put everyone together, it's possible.
I don't know if it's in time for Mila. It is too early to know that, but I'm really excited that we're in a day where it looks like there's hope.
Milasen remarkably stopped and even improved a number of Mila's symptoms in the first year after she began receiving it. But in the second year, it was unclear whether or not it was able to fully stop her symptoms. And by the third year, it was clear that her disease was progressing and her quality of life was not at a place that I had ever hoped for my daughter. I was forced to make decisions that no parent should ever have to make. And in February of last year, Mila's big spirit left her little body. And I am learning to live every day of life now without her physically here with me. You know, when I first started fighting for Mila, right when she was diagnosed, I had no idea that her story would take this path that would end up in her becoming the first person in the world to receive a drug tailored to a single person. After she began receiving Milasen, I started learning a little bit more about what did individualized medicine mean. Some people call them N of ones. You know, and I started learning that individualized medicine was something that was very new. It hadn't been done before. And it was a new approach to treating disease. Traditionally, drugs have targeted diseases or conditions or genes. In Mila's case, what it showed was that a drug could be designed specifically to target the underlying genetic cause of the disease. In her case, it was a single mutation that no one else in the world shared. So in rare disease, what happens is there are very few cases where there are mutations that are shared amongst many different people, but that's kind of an exception. And what generally happens is there are many, many different mutations within one gene or one disease. That was the case for Batten. For example, in Mila's type of Batten, CLN7, if you look at a database, there might be 45 children and 43 different mutations. So inherently in rare disease, by targeting an underlying mutation, there tends to be just one or a handful of people that share that mutation. And Mila's mutation happened to be amenable to an antisense oligonucleotide, or an ASO, that was similar to Spinraza. Spinraza was an incredibly successful drug for the disease spinal muscular atrophy, and it was approved just around the exact same time that Mila was diagnosed. So today I want to talk about the risks and the challenges and the hopes, and how that's a little different for the individualized medicine approach. You know, for the risks, the risk-benefit analysis was different for Mila. She was the only person in the world that would ever receive Milasen. And so the way I thought about it was that the risk of treating Mila versus the risk of not treating Mila. And that was a little bit different in how we were thinking about the risk-benefit analysis, which is kind of the base of the house that you are building on. The risk of not treating Mila was very black and white. She had a degenerative fatal disease, 100% fatal. So the risk of not treating her was black and white, in that she would lose each of her abilities and she would die in the next few years. The risk of treating Mila was that this was a new drug and it was unknown, and it looked very, very promising in her cells in the lab. It was restoring health to them in a pretty unprecedented way. But it was still new, and it was designed just for her, and it hadn't been used in other humans. And Dr. Yu did a very good job at setting expectations very correctly, at explaining that it looked very promising in the lab, but we didn't know. We did know that this type of approach was very successful for spinal muscular atrophy. But that was a different disease. It's a disease in the spinal column, and in Mila's was throughout her brain. And so that drug had to travel up the spinal column and penetrate all of her cells in her brain, which was different than spinal muscular atrophy. So the expectations were definitely set correctly, but I didn't spend more than a second thinking about whether or not it made sense to do Milasen or not, because the risk of not treating Mila was so much greater than the risk of treating her that it made sense. And very good science had gone into showing that it looked as safe as possible and possibly efficacious. And so that was kind of the decision-making behind the risk analysis. In terms of the challenges, I think the biggest challenge was that our current system is not designed for individualized drugs. It was designed for one drug for tens or hundreds of thousands of people, not tens of thousands of drugs for one or very few people. So what does that mean? It means that there's not an appropriate or proportionate regulatory path. There's not a clear regulatory path. Milasen fell through the cracks, and the FDA did a really amazing job at responding to it in a very rapid and safe way, and allowed it to happen even though there was no precedent for it. Another hurdle is that there's not incentives for investors and insurance companies or Medicaid, because they're not used to this paradigm where now suddenly there are potentially in the near future many, many drugs, each targeting one or very few people. Another problem is that there isn't a place to gather the data that's learned from children like Mila, the preclinical and the clinical data. All of that data, if it remains in silos with each doctor that's working on a case, we'll never learn and we'll never be able to scale this. So having a central place where all of this data sits to learn to be able to scale it is fundamental, and being able to have more people like Dr. Yu learn how to design and create and get approved, drugs like Milasen was also not, was a very big challenge. So I've tried to play a role together with Dr. Yu and many others in trying to face these challenges. One of the things we're doing is we're working with the FDA, who has issued 4 guidances on individualized medicine in the last year. I'm very happy to see that they care about this. And we've had ongoing discussions with them for a number of years, and they do care, and they realize this is the future. I think they realize that a decent percentage of medicines in the near future could actually be individualized medicines. And so we need to start adapting and changing the system that is not equipped for this right now. But we do need to, and we are working with them and collaborating with them to come up with a more appropriate path for this paradigm, this individualized medicine paradigm. In terms of the business model, reimbursement model, why does it matter? It matters because if we don't have a business model and there is not reimbursement, there won't be access, and this will trickle through academics. We need a much more sustainable model. And so I do believe that discussion is being had right now. I'm part of those discussions as well, but it is kind of secondary to the regulatory path. We need to have a correct regulatory path that's appropriate for this, so that we can then continue the discussions with private insurance companies, with Medicaid, and with investors, so that we can create a business model that's viable and that can take this forward and create a sustainable individualized medicine ecosystem that's not reliant on academics only. In terms of the data and gathering the data into one place and having really transparent data for anyone in this field so they can learn from the good and the bad. Together with Dr. Yu and a number of others, including the Oligonucleotide Therapeutic Society, which is kind of central to ASOs, we started the N-of-1 Collaborative last year, which is kind of a hub for primarily scientists, physician scientists. And the idea of it is to create a toolbox of how to do this. How do you create drugs like Millicin? Because there's only 1 or 2 or 3 people in the world that have done this. And so the idea is to create, how do you choose the right patients? How do you design and screen ASOs? What does manufacturing look like? What does safety studies look like? How is the trial and outcome measures? How are they designed? We're working on all of these aspects right now, and there's about 150 members from around the world that are working on this. We are also creating a database, and the idea is, Mila's data will be the first one to be included, is that all the data will be dumped in, ideally both preclinical and clinical, so we can learn. And as with time, the idea would be is any academics and ideally companies, although that might be idealistic to say that, would be dumping in this data so that we can learn. Otherwise, we risk not being able to scale this and being stuck. And the idea of the N-of-1 Collaborative is to grow many, many more TIMUs that are able to do this and that have their toolbox and they have a place to put their data to share. So this now becomes something that is happening in academic centers and companies kind of around the world. In terms of the hopes, the hope for me is, the way I think about it is like this. Rare disease, especially in children, is a global health crisis. I'm always shocked that people don't use stronger words. There are 60 million children that will die before the age of 5 from a rare disease. That is the population of Italy today. Every single human being in Italy is a child that will die from a rare disease before 5 years old. If we go gene by gene, it's just not going to cut it. I'm glad that we are going gene by gene because that's better than nothing, but it's not enough. So having kind of a platform cross-cutting approach, which is something like individualized medicine offers, it offers a potentially incredibly impactful solution to this global health crisis. Now, we've just identified the challenges that we need to overcome to get there, and we're working on them. But my hope is that individualized medicine, starting with ASOs, that's the modality right now. And to be honest, we're really only using one type of ASO right now, which is primarily focusing on splice modulating, which only a percentage of patients are actually amenable to something like that. Maybe 5 to 10% of patients might have a mutation like Mila's that's amenable to a splice modulating ASO. But we're learning more about allele and gene knockdown ASOs, and they are happening there in the pipeline as well. There's a little bit more to learn about them. There are also other types of RNA therapeutics. There's gene editing. So there'll be more and more tools in that toolbox for individualized medicine approach. And my hope is that in the near future, that individualized medicines has its own path, regulatory path, its own path to reimbursement, its own path to having the whole ecosystem of families and patients and scientists and academics and VCs and companies and insurance and regulators and the NIH, and everyone kind of has changed their mindset to imagine that a decent percentage of treatments will be individualized and that this offers a really, truly impactful solution. And it starts helping hundreds of thousands with time, maybe more. And, where I end up, being stuck wide awake at night and can't sleep is I think we've treated Mila, but now how do we get from Mila to millions?
Right. What an incredible place to end with our talks. How do we get from Mila to millions? So Julia should be able to join us virtually. So while they patch her in, if there's any questions in the audience, we'll open up for our panel discussion now, and then we'll go from there. And while everybody's getting kind of set up, welcome, Julia. Can you hear us okay? Wonderful. Okay. So, thank you so much for your talk, and thanks for doing that even while you were sick with COVID. So we appreciate you trooping through that. So we'll get started, I think, with a question to Pat, if that's okay. So how can patient advocacy groups support families who've participated in gene therapy trials, especially since we're thinking about that natural history is changing, right? Our medicines are getting better, but still may not be perfect. And identifying those new issues that can creep up, and how can advocacy groups play a role in that?
Sure. Well, first of all, we're in early days, but what I think what we need to do and what we're trying to do is these growing number of families are creating a list of questions that they have. So we have, and those questions are categorized across the standard of care. So for instance, how much exercise is too much exercise with a microdystrophin on board, which is an unknown? How will we get there? We are looking at expanding an MRI, muscle MRI study with University of Florida. So these children get in systems and processes so that they can be evaluated over time very systematically, and so that we can design protocols and looking at a master protocol for these children so that as new therapies come online, they can be in a study that we can look quickly or at least look thoroughly to see if this, for instance, in an antisense oligonucleotide, if someone's amenable, we have 4 approvals in this country, exon 53, 51, 45, and 253s. But if this child is amenable to an exon 53 skip, should that child be have that antisense, and should we look at that? Is it going to be synergistic? Is it going to be complementary? How are we going to look at that? How are we going to know what's beneficial? So I think having a rigorous, we've talked about post-marketing studies, we've talked about long-term extension studies. I think those long-term extension studies have to be with the forward-looking thought that we are going to implement new medicines to them as they come online, as they seem appropriate, and study them so we can know if we're protecting that benefit and extending it over time.
Thank you. I think you kind of touched on another question that has kind of come up through the day too of if there are multiple options available, how do families choose which is the right trial for them to participate in, especially with potential other options coming on the near horizon with as quickly as things are changing. So I'd love to hear from Pat, of course, but I think also Dr. Sade, I think you could kind of speak to how you, on the physician side, can talk through those discussions too.
Sure, I can start off. The gene therapy studies have very tight inclusion criteria, as was described in earlier presentations, because you want a cohort that's fairly stable and rigorous in terms of understanding what that particular group of people with similar characteristics how they're going to respond to that therapy. So if you're young, 4 to 7, gene therapy is a consideration. And we're talking about also antibodies to the virus, so that can knock you out of that gene therapy study. So I think what most physicians are likely to do is really spread out the opportunities for these families. They're always on standard of care, which is steroids and some cardiac meds typically early on. So then decide what can we do and talk about the benefit and risk with these families. Because as we have seen in the MTM studies as well as Duchenne muscular dystrophy, we've now lost patients, lost people in those studies. And so there is real risk, and it's on the table to be discussed. It's one of those elephants that someone died. How did that happen? What do we know? What did we learn? So I think families need time. Time is the commodity on all of these rare diseases because time means loss of function. Time means everything to these families, and time from the physician community and their teams to sit down and go through it repeatedly. Because sometimes you hear a few words and then it goes to a hum, right? Because it's just too much. So to keep going back and say, these are the options, and do some serotyping. Look and see if this is options for this child. Because if he's got antibodies to the virus, then other options are going to be on the table. So I think it's being thorough, considering what tests and what information can you give, what tests can you do to really encourage the discussion and make it more reasonable to the families. Here are the options. If they close down, then we can look at other options. So I think it's really about time, trying to get as much information as possible and trying to have repeated discussions.
Thank you. This comes up a lot, increasingly so recently, more so with newly diagnosed patients. So I had a newly diagnosed child in clinic who is eligible or who's amenable to an FDA-approved exon skipping therapy, and who was just diagnosed. And we had talked about the diagnosis, starting steroids, standard of care, cardiac prophylaxis, and all of that, and then went on to talk about exon skipping, and also said, by the way, there's gene therapy on the horizon. There's all the hype and excitement around gene therapy, but the following concerns and risks that are very real, and there's no guarantee. The specific example that I have in mind is a child who was diagnosed late, so 8 years of age at the time of diagnosis, and so would not have been included in the initial cohorts for the current gene therapy trials that plan on enrolling younger patients first. And so we had this discussion of, do you go home now on only steroids and hold off on starting to receive exon skipping therapy, which could have cumulative added benefit, with the hope of maybe you may be eligible for experimental gene therapy. And so that's a very long discussion that we normally have, and it remains open at the beginning, but we do start the start form for, and people may not all be familiar with that, but if you're amenable to exon skipping, most of the time that's not something that your physician can prescribe and you get the next day. There's prior authorization work that has to come into place. There's potential finding home nursing or infusion sites and all of that. And so I think with this specific family, we talked about that and had them come back for follow-up soon after to rediscuss everything in person, and they had opted for exon skipping therapy. And then one thing that I also make sure to add is that based on the current knowledge and evidence, it does seem that treatments there's going to be cumulative benefits from several different treatments and not a one and done type treatment for children.
Julie, I know you're virtual, so I wanted to give you a chance if you had any comments on that section. Not hearing well.
Yeah, it's pretty hard for me to hear my best.
Okay. Yeah, so we were just talking about how patients can think about navigating the different options that may be available. And I do remember with Mila in particular, there was a gene therapy being developed, but also the ASO that you guys were working on. And I know I see this in my personal experience working with different foundations and kind of helping families and advocacy groups guide their communities through these different options and understanding kind of the pros and cons. And if you have any thoughts on how to have those discussions or parts that were really hard or parts that you thought could have gone better even.
Yeah, it's very, very hard for me to hear you. So if I understood correctly, is when you're faced with, when a patient or a family is faced with more than one treatment option, is that correct?
Yes, when you've got multiple different options, I think they turned my mic up. When they've got multiple different options on the table, how do you navigate that, especially coming from a place of you don't necessarily know the science behind them?
Yeah, and I'm sorry because I can hear like 1% of what's being said. So I can share my own personal experience if that's helpful. While I started the work towards a gene replacement therapy for Mila's type of Batten disease, in the meantime, the individualized ASO came up, and that's the path I ended up following. People from both of those teams agreed that it was going to be faster for Mila to access the individualized ASO and very possibly more efficacious. So I kind of asked all of the different people involved, if this were your child, what would you do? I asked that to so many people and I started getting the same answer. Because there's a lot of unknowns right now. I was heading towards Millicin, thinking that it was a bridge to gene therapy. And like all of us probably experiences, things morph and change. You start learning. And the ASO looked, the more and more, we looked at how her cells were responding in the lab, which was pretty amazingly. It started actually looking like that was going to be her very best shot first. Timing wise, the gene therapy was not going to happen in time for Mila. It was taking years, and it was very obvious that that was not going to line up with her progression of her disease. So that was also taken into consideration. And then I will say that as I learned, I had first learned about gene therapy, and everyone had said one shot and you're done. And that's
Julia, I'm sorry, we lost your audio here. I don't know if they still have it online. No, not yet. I think they're working on it. I think they were trying to fix an echo.
let me know if you can hear me now.
You're back now. You're back.
Okay, I didn't change anything, but okay. The last thing I was saying, I think where it cut off was that I had seen the one-shot gene therapy option as a positive thing, but as Mila's disease progressed and I headed toward an ASO, the concept of having administration every few months, which ended up being extremely minor, was significantly less impact on her than going to the dentist. And I'm actually not joking. Going to the dentist with Mila was like an hour that was horrible. And having Millicin was like a 10-minute procedure where she didn't have anesthesia. We no longer had to travel because we eventually moved it to Colorado near us. And there's a lot of waiting around and stuff and tests for a number of hours, but it was like a 10-minute procedure that was very, very easy. She didn't flinch. And so for me, doing that every few months meant that if I wanted to, I could stop. And to me, I didn't know that going into this, but it ended up being extremely important for me. So the idea of having a one-shot deal was out of the question for me at a certain point with Mila. I wasn't going to potentially freeze her in time in a state of limbo. And so there is a great place for that, but I moved out of that place. And so the idea of an ASO became more, it made more sense to me. So those are some of the decisions that I don't know if that's helpful, that I thought about in the process.
I think that was an incredible way to process through that and help others think about it, that it's also, there's no right or wrong answer for a single patient. Everybody's journey is a bit individualized, even if we're talking about the same treatment for multiple patients. I think it segues into a question that often comes up, is talking about gene therapies inclusive of RNA-based therapies or even CAR T as a cure. And we'll sometimes say it's a 4-letter word that we don't use. Sometimes you use it and say, maybe it could be. But I think often we're thinking about what are the other treatments that are going to come or need to be added on, and not to think about that in terms of a clinical trial, but I'm just curious about the panel's thoughts and how you navigate those discussions with your community partners and your groups of, is it a cure, is it not a cure, is it a therapy, how do we talk about that?
I'm happy to jump on. Previous to coming to my current position, I worked closely with the SMA community, and we did call it a 4-letter word, the C word, the 4-letter word, because what had been shown in clinical trials was that depending on how far the disease had progressed, that really impacted the outcomes. So therefore, in SMA especially, and in many diseases, newborn screening was really, really important to catch kids early with treatment. However, there was a lot of hope within the community that gene therapy would be a cure. And we spent a lot of time, learning in partner with our advocacy groups to understand, one, what was the community thinking, but then also how can we address this concern? So we worked not only in our presentations, I think it was mentioned earlier at advocacy symposium meetings, to say this is not a cure and really manage expectations, but we also partnered with the advocacy group who was aware of the data and the researchers in the area who could, again, help manage expectations with the communities that we're working with. So, and now stepping into where we're at today, I feel like there's a little more acceptance, but there's still that hope from families. So again, we expect that we do our best to manage expectation in partnership with clinicians as well as our advocacy partners.
We did some work with our community about what, excuse me, what does the word cure mean to you? Because everyone has a different definition of what a cure would be in terms of their own child. So cure in some cases meant normal. For the little boys that were used, this word was used, they believed that meant exactly like my friends. So we found that the word is really difficult for children to understand because of their assumptions associated with that very simple word. So we tried to get their parents to understand that this is a disease that you're going to have all of your life. And what we're trying to do with these therapies is halt disease progression, put that pause button, push that pause button for years and hopefully for a lifetime. But most of these children are born SMA1 as well. If you don't treat in the first days of life, you have considerable damage. So the change that this child is going to undergo is not going to be full cure, but it's going to be dramatic in terms of their quality of life. So I think this goes back to language, about how we use language. And I think for these patients and families, there's also always a risk-benefit discussion on here about the AON therapies are weekly infusions. How does that look? How does that look in terms of benefit? And what will that do? And then can you couple that with other therapies? So it's a really tough word to use.
At least from my perspective, it's a no word as a physician. I do not, the reason we do what we do is in hopes of bettering children's lives and helping them and approaching an ultimate cure, but that's never something that I use, especially because different people hear different things and then could have hopes, disappointment, frustration, and may overlook other things that may help them as well because they think, well, they got the cure already. So I don't use that word at all. I go into a very lengthy and often repeated discussion because people don't hear everything the first time you talk about it, myself included. So I talk mostly about risk and benefit and what could help with every family given their personal circumstances and individual choices.
I would love to jump in, and I'm so sorry if I'm repeating anything that the three of you just said because I cannot hear words, so I'm sorry. I think it's really important to head towards a cure. I think the cure is the ultimate goal. However, setting expectations correctly, I can only go off of my own experience and the experience of others that they've shared with me who have received treatments, is that the reality is that this wave is rising and it's really exciting, but it's early days. And while it could be incredibly curative for some people, for many, I think you will be in limbo. So the way I described it for Mila was the way Dr. Yu actually described it very, very well, actually, for me. It sticks in my head from before Mila's began was, this is for a degenerative disease. Some of Mila's cells had died. Some of them were in the process of dying and could be stopped. Some of them were in the process of dying and couldn't be stopped. And so there was this, that's kind of what I saw, is that Mila had so many different things going on in her brain and so many different symptoms. Some of them were stabilized and improved. Other ones weren't. And so there is this limbo that you're in. And I've heard that from many other families as well that have children that are in treatments. So while I hope that it's more and more towards the curative side over time, and it will be for sure, limbo is kind of I think, perhaps a good expectation to set for all patients. And if you supersede it, great. That's just how I think about it.
Oh, nope, there it is. Sorry. So I think those were all phenomenal answers, and it's a really touchy, tough topic to discuss, and really personal. I think we've got just about 5 or 10 minutes left. So I want to give each of the panelists an opportunity. If there's just something that's kind of burning on your heart and you want to share and we haven't gotten to touch on it or talk about it yet, and maybe we'll start with Pat and move our way down the row.
For me, it's language. It's what we say and how it lands and how we have to be really thoughtful about speaking to families who have children who have catastrophic, complex, multi-system disorders. And I think that we all want to, as Julia said, we all want to push toward cure. But for the people living today who are not going to be eligible for these amazing therapies that are coming online, we can't give them nothing. We can't write them off. We have to always be mindful of what we can do, for who we can do it, and to be able to take time to discuss those benefits and risks and hope for the best for these patients. It is so hard to look at a future when today matters most.
And I'm going to expand a little bit on what Julia started talking about. Is I think now is the time for all parties involved to get together, share knowledge, both positive data, but also side effects, bad outcomes, fatal, tragic outcomes as well, and use treatment modalities to develop that therapeutic toolbox and make it so that information is not siloed and that we could help patients across trials and across diseases as well.
I would just say, loud and clear language is so important, and the only way to do it is, like you had mentioned, is us all working together from an industry perspective, clinician perspective, and advocacy perspective. So I'm very passionate about engaging early, engaging often, and given that most of the people here and on the line are with advocacy groups or caregivers themselves, if you're not engaging and you want to reach out, I think hopefully industry partners, I think what you heard today is they do want to listen and they do want to learn. So we can come up with common language, we can learn from each other to move these forward as quickly as possible.
And can you hear me okay? Okay. What is on my mind here at the end is not a beautiful grand finale at all, but something kind of more technical. What I want to make sure is clarified for anyone listening, because it's the number one question I get on a daily basis from families, which is, when does an individualized medicine make sense? I don't want to tell this whole story about Mila and about the future of individualized medicines and not kind of clarify that. I think that many people understand the individualized medicine path as another alternative to a trial, a proper trial, and that's not actually the case. The best way to describe it is Mila was the only person in the world that had her very specific mutation, which Milasin happened to target, which was designed, to target. And the way I would describe it is that if the underlying genetic cause, which is often a single mutation, happens to be shared by 1 or 2 or 4, or there is not a specific number, a very, very small group of people. And it happens to be, that mutation happens to be amenable to a modality, which the one that's on the table right now, which is most programmable is ASOs. There are different types of ASOs, like I mentioned. If an ASO can target a mutation, and there happen to be a very small number of people as opposed to hundreds, for example, which is more of a proper trial, or even 50 or 30 could be a much larger group of people. If it's very, very small, then that is the new kind of regulatory path that we're trying to create. following kind of Mila's. And there's been a handful of children who have been treated with an individualized ASO since Mila began Millicent 4 or 5 years ago. So that's how I would describe it. Another way of putting it is, if you're thinking about the modality on the table right now, so splice modulating ASOs, like I mentioned earlier, there's a small percentage of mutations and rare disease that are amenable to that. I happened to figure this out because Mila was missing a mutation, and she has an autosomal recessive disease. So there was one found. Mom and dad both have to pass them down, and there was one missing. When one is missing, it's very often that it's kind of an intronic mutation that no one can figure out. And that happens to be often, not always, targetable by a splice modulating ASO, so that's the only way that I can tell you I've experienced that. So not everyone falls into the category of having to have a mutation that happens to be shared by only 1 or 2 or 4 people in the world. With time, this will grow, like I mentioned. So as there are other modalities like allele knockdown and gene knockdown ASOs, and like I mentioned, RNA therapeutics and gene editing, there will be more tools. That will be more and more and more people will be amenable to this individualized approach. But it is still really new, and it is not a path for everyone. Now, should you assess it and evaluate it? Yes. The question I'm given all the time, which is very hard for me to answer, I can only tell you I'm working really hard with others to have a better answer for it, is that, Dr. Yu is doing the best he can with hundreds of inquiries coming in. 10% of them maybe are amenable. And Lorem is doing a good job at trying to field inquiries as well. There really is no great place, but I can only tell you that we're working very hard to make sure that, like, the N of 1 collaborative is empowering many, many more people out there across the world to be able to assess and be able to design and be able to treat more children. So it's still early. I wish I had better answers, but that is the question I get from most people. So I hope that helps clarify. And thank you again for inviting me. And I'm sorry I can't hear your amazing answers.
Thank you so much for logging in for us, Julia, and thank you for everybody who stuck with us all afternoon. I think that was a great, hopeful place to end. I hope to see more symposiums like this at more conferences, not just in our echo chamber of gene and cell therapy, but get them out into other platforms too. And again, thank you everybody for attending and for participating. And don't forget that we have our reception to follow here starting just in about 2 minutes. It'll be on the 3rd floor of the convention center here, and everybody is welcome to join. Thanks so much, everyone.